Good morning, everyone. Goeiemorgen. Thanks for joining us this morning for the last day of Medecos 2022. Just want to check if there's anybody else. If there's anybody else outside who wants to join us, please move into the hall. Then we'll get started. This morning we have Kerstin Braun, who's going to give us a keynote address. I'm not going to say anything else because she said she's going to introduce herself. So thank you very much. We look forward to your talk.
Okay. Yeah. Well, good morning, everyone. First of all, I wanted to thank everyone who made it here at 8 a.m. on a Friday morning on a 5-day conference. So thank you very much. Yeah, I think there's not that much of an introduction for me. But yeah, my name is Kerstin Braun. I'm an assistant research scientist at Arizona State University in the United States. And I wanted to start out with a little disclaimer. I'm a geologist, so this is not going to be talking about anything related to vegetation today. But what I wanted to talk about is the research that I've done on speleothemes, which is basically a fancy word for stalactites and stalagmites, so cave deposits, and what it might tell us about diversity in Mediterranean climate regions and its evolution over time. Does this work? Which direct? Okay, this is not. This is not the right one. I pushed this button. Is that the wrong direction? I think this is the wrong direction. There we go. I was holding it upside down. Okay, so yeah, in general, when we are thinking of global patterns of plant biodiversity, here shown as a kind of colorful maps of vascular plant biodiversity by area. We often think about this gradient from the tropical regions, here shown kind of with higher diversity in these purple and bluish colors, towards lower diversities in the polar regions that is indicated by the kind of more yellowish tan colors. And we've kind of tried to explain these patterns mostly by through metabolic theory, which states that in warmer climates, plant metabolic rates would be increased, which of course then increases genetic divergence. Or things like water energy theory, which I think is more based on just an observation that in general, when we find a climate that has higher temperatures and good water availability, that's where we see higher diversity in the plants. But here comes the shocker to everyone in this room. Of course, the Mediterranean climate ecosystems are kind of an excursion to this trend from the tropics to the polar regions. And especially when we're thinking about the climates, they're dominated by winter rainfall. So both metabolic theory and water energy theory, of course, can't explain these high diversities that we find in these regions that are dominated by cool season main growth periods. And here again, just to show how special the Cape is in this respect. So this is a graph that Adrian showed on Monday, showing the residuals of the species area regression model. So basically showing that for its area, the Cape Floristic region has quite high diversity. And in this case, it is compared, of course, to different tropical biomes on Earth, shown kind of in the greenish and bluish colors, and the Cape is shown in orange. Now, in the Cape specifically, I think this has been tried to explain in the past using environmental stability theory, which basically states that historical factors in climate and topography can also affect diversity and its evolution. So basically, if we have constant but moderate diversification rates, but low extinction rates because we have a very stable environment, then that can over time lead to higher diversity. But so far we don't really have a paleoclimate record from the kind of diversity center of the Cape Floristic region that shows this kind of climate stability. So what I want to do here is show you one of the first really long-term record that covers multiple glacial, interglacial timescales that we have from the kind of western part where we have the highest diversity in the Cape. And then I want to compare that to records from other Mediterranean climate ecosystems. And I'm using speleothem proxies again for this, stalactites and stalagmites from caves. And the question that we're asking then, of course, is do these suggest that there is a more stable climate in the Cape than in other Mediterranean climate ecosystems? But I'm also going to make a comparison between the winter rainfall-dominated western parts, where we have the highest diversities, to the more eastern aseasonal parts of the Cape Floristic region, where diversity is somewhat lower. Okay, so I guess most of you are not very familiar with using speleothems as paleoclimate archives. In the most general sense, for the formation of speleothems, what we need is a decent amount of rainwater. In a winter rainfall climate, we think about 300 millimeters per year is a good amount. And then we need at least a little bit of soil cover with some vegetation. So what happens is when the water infiltrates, it encounters the soil atmosphere that is enriched with CO2 due to the decomposition of organic material. And that way, the CO2 gets dissolved in the water and makes it slightly acidic. And that allows the dissolution of a carbonate host rock. And then as the water infiltrates further through the karst, it at one point or another encounters a larger cavity in the rock. And that the atmosphere within these cavities usually has a lower CO2 concentration than the soil atmosphere did, which leads to the degassing of CO2 and also the precipitation of the carbonates that have been dissolved previously from the host rock. So that's kind of a simple way of explaining how speleothems form. And the big advantage that we have with using speleothems over other paleoclimate records, such as lake and marine sediment cores or ice cores, is that we can directly date them using uranium-thorium methods. We're now able to do that up to about 700,000 years ago. So, just a few quick words about the methods that I'm using. I think many of you know what an isotope is, but I thought I'd just mention it quickly for in case some of you don't. So we use carbon and oxygen from the carbonate molecule, or calcium carbonate. And these isotopes that I'm going to talk about basically have different numbers of neutrons in the atom. So that causes ever so slight differences in the mass of these different atoms, which leads to fractionation during different processes. So for carbon, of course, biological processes can be really important. And then for oxygen, processes on a water cycle are often what causes fractionation. And the way that we report these is shown with this beautiful little equation there. We call it the delta notation. And basically, we just measure the ratio of the heavy to the light isotope in our sample. And then we standardize it with an international standard material. And we report it in per mil. So again, coming to how we interpret the speleothem oxygen isotopes. In the most general sense, these are kind of driven by temperature and water cycle processes. So things like evaporation, water moisture transport, sometimes the mixing of different air masses, re-evaporation of terrestrial water can all influence are oxygen-stable isotopes. And from the research I've been doing here in the Cape, we found that on glacial interglacial timescales, mostly in the eastern parts of the Cape floristic region, it is very likely that these changes in oxygen isotopes are caused by changes in rainfall seasonality. So during times when we have more summer rainfall, the oxygen isotopes would be higher, and when there's more winter rain, they would be lower. But here in the western parts of the winter rainfall region, most of the paleoclimate records that we do have, as well as modeling, indicate that there isn't as much of a change in the seasonality. It's more likely that we see actually an increase in winter rainfall during glacials. And because of that, we think that in this region, temperature and the composition of the oceanic source of the moisture might be more important than actual changes in the seasonality. Okay, coming to the carbon, I think some of you might be a bit more familiar with this, but for speleothemes, in the most general sense, local vegetation and soil processes are kind of the driving factors for this. And then, of course, there's an impact of what kind of organic matter we have in the soil, and that depends on the vegetation. So the proportions of C3 and C4 plants can have an impact, which of course is mostly important in regions where these plant types might overlap, which we do have in the eastern parts of the CFR a little bit. Things like forest versus shrubland versus grassland vegetation can also affect our delta 13C. And then there's a whole range of different indirect effects of water availability that can have quite an impact as well. So the vegetation density, soil bioproductivity, and this process we call cryocarbonate precipitation. But all of these usually should increase the delta 13C when it's drier and decrease it when it's wetter. So just to summarize this a little bit, when we're thinking about speleothemes as archives of vegetation, I think the stable isotopes can be really useful because when we have changes in the oxygen isotopes, they often reflect changes in the water cycle, water availability, and temperature. And of course, if these changes are large enough, they will affect vegetation as well. And then the variations of the carbon isotopes usually reflect changes in the vegetation itself or in soil processes that are either related to vegetation change or cause vegetation change. So I think these are pretty good proxies for this. Okay, so I'm going to talk a little bit about the site that we have our record from. It's called Robertson Cave. It's in an active quarry that is operated by Cape Limeworks, so just outside of the town of Robertson, and the location is shown on the map in the bottom here. The current climate there is relatively warm and dry, so it's about 17 Celsius with a mean annual rainfall of 300 millimeters, and most of that falls in the wintertime. So when we were there for sampling, there was actually no dripping and no active deposition of speleothemes in the cave because we were kind of just at that boundary where we get enough infiltration to deposit speleothemes. I also wanted to quickly mention what we do with these samples after we take them out of the cave. So the first thing we usually do is we cut them in half with a saw along there. We try to get as close to the main growth axis as possible. And then in this case, I'm showing one of the samples that we have from Robertson Cave in a scan on the left with its outline and some indicators of where I sampled it right next to it. And you can see a little blue line that kind of goes from the top all the way through. That is where I sampled for the stable isotopes. I think the individual sampling points are a little bit hard to see on the scan, but it's basically little dots that are kind of offset a little bit next to each other. And then there's some larger shapes that basically mark, that have the letters and numbers on them, and those are basically the locations where I dated the sample. And all the dates are listed here as well. So the graph on the right shows the depth in the sample on the y-axis and then the age on the x-axis. And this is basically what we call an age model. And we have multiple different kind of statistical ways of constructing an age model like this. I used an algorithm called Bacon, which you can use in R. And basically, you know, the graph just shows all the individual ages that we measured, and then we interpolate with these statistical methods between them to get actually an age value for each one of the individual isotope analysis. Okay, and what we end up with in a cave like this, where we have 5 different samples, is a whole bunch of different wiggly lines. So in the top here is shown the oxygen isotopes, then in the middle the carbon isotopes, and in the bottom I have the ages with all the errors for all the individual samples. And as you can see, this sample grew from about, the combined record basically goes from about 200,000 years back to about 660,000 years. And the numbers in the top here indicate what we call marine isotope stages. So the odd numbers there will be warmer intervals, and the even numbers will be cooler intervals on a global scale. I guess the other thing I wanted to say here is that when we look at these records, we can see that there's quite a bit of noise in them. And there are some parts of these records where we do have quite a good overlap between the different records from different speleothemes within the same cave. But in some cases, there's quite a bit of distance between them. So for the analysis that I'm doing here, where I'm basically going to use the amplitude and the dispersion of all these records to make an assumption about climate stability versus climate variability, it is really useful to have multiple records from the same cave that also overlap in time. The other thing I wanted to mention, that this time interval that we're covering here is quite old for speleotheme records. We don't have a lot of records that go that far back in time. Most of the speleotheme records, probably due to reasons of preservation in caves and things like that, cover just the Holocene, maybe going back into the last glacial. We do have a few longer records than that, but the vast majority will probably be within the last 100,000 years. Okay, so what did I do with this? As I mentioned, I wanted to compare this record to records from other Mediterranean climate ecosystems. And then again, because there aren't a lot of records that go actually back as far in time as our record. So basically, I made the assumption that if a record from a different Mediterranean climate region covers one time interval that is considered by paleoclimatologists as a full glacial and one full interglacial, that basically represents the variability that we get on glacial interglacial timescales. Sorry. Then, of course, I'm looking at Mediterranean climate ecosystems, and I use the Köppen-Geiger climate classification to find places that are currently in Mediterranean climates. So I use the CSA and CSB classes for that. And then, of course, I also did a comparison between the winter rainfall-dominated CFR and the year-round rainfall-dominated parts in the east. And for that, I basically used some records that I have been working on over the last decade that were previously published. And to kind of statistically make this comparison, we used Flickner-Kailin tests, and we thought that those were really useful, mostly because they can deal with data sets that have very different numbers of data points, but it also deals well with non-normally distribution. And we, especially for the Delta 13C, we often had somewhat skewed or bimodal distributions. So that's why we went with this test. Okay, so here is basically our map of the Köppen-Geiger climate regions. And as you can notice right away, we don't have any records from the Mediterranean climate regions in South America and Australia that fit these criteria that I used for record selection. I have 2 records from California, and then the records from the Mediterranean basin are kind of grouped into different regional groups. So I have 1 record from Portugal, 2 from France, 2 from Mediterranean islands, 1 from Italy, 1 from Turkey, and I think 5 from the Levant. Okay, so just quickly showing the results. So these are basically the outcomes of the Fligner-Kylien tests. So what these do basically is they show you a ratio of the dispersion of the two of the comparison record to the Robertson record. So if this number is close to 1, then that means the dispersion is similar. If it's above 1, it means that the dispersion at Robertson is lower than the record we are comparing it to. And what you can see is that quite obviously all the Northern Hemisphere records from the other Mediterranean climate regions appear to have quite a bit higher dispersion than what we find at Robertson for both the oxygen and the carbon isotopes. And then I'm just showing it quickly here as well as box plots, so you can just get an idea of the total amplitude that we see in these records. And it's also showing, so Robertson is here on the left, and all the other regions are plotted kind of towards the right from there. Okay, so, and yeah, so now I'm coming to the comparison within the Cape Floristic region. I'm using for this 2 records that are, or 2 locations that are on the coast. One of them is Pinnacle Point, which I actually have 3 different cave sites from. And then I have 2 records from inland. One is from Kangoo Cave that was published by Talma and Vogel in 1992. And the other one is a record that I published from there. And of course, all of these are, so this is a vegetation map, and all of them are located in what we consider the Cape Floristic region. Now, climatically, they do differ quite a bit. So again, Robertson Cave is kind of in the west, currently gets relatively little rainfall, about 300 millimeters. The sites further to the east, especially the coastal sites, do get quite a bit more rainfall than that. But also the inland sites, I think, get as much as 500 millimeters, mostly because they're in the foothills of the Swartberg Mountains, which is a mountain range that's, I think, in this area about 1,500 meters tall. So that causes the rainfall there to be a little bit higher than in the surrounding areas. But more importantly, of course, what we see here in the map on the right is that the seasonality of the rainfall is quite different. So at Robertson, we're in the winter rainfall area, whereas the other sites are really a seasonal rainfall, maybe with some peaks in the transitional seasons. And this, of course, also is reflected in the diversity of the flora. Sorry. Okay, so yeah, these are just 3 plant groups that are kind of characteristic for the Cape flora. Erica, Aspalathus, and Proteaceae. And of course, we can see that the diversity centers for each of them is very close to where we have our Robertson record, and the species numbers go down as we go east. Okay, so I added these to our Fligner-Kylien tests. And what we can see is for the carbon, both the inland and the coastal records do show quite a bit higher dispersion than the Robertson record. Whereas for the oxygen, we do have kind of a similar value to the Robertson record at the inland sites, whereas the coastal records again have higher dispersion. Now, if we look at the box plots for this, again, Robertson is here on the left, and the south coast and inland records are towards the right. We can see that, you know, with if we consider the somewhat outlier values in the inland record, there is quite a bit higher amplitude to the absolute values in that as well, even though the dispersion didn't seem to show that as good. Okay, so already at the conclusion. So I just presented to you a new record that we have from Robertson Cave speleothemes, and this is one of the first long paleoclimate records that we have from the winter rainfall area of the Cape Floristic region. So we don't have really any records that go much beyond the last glacial. The comparison with other Mediterranean climate regions shows that the climate, or indicates that the climate was probably more stable in the western Cape Floristic region than in other regions. And the same thing was found for the comparison between the eastern and the year-round rainfall region. So I think that this might show us that environmental stability theory does kind of really work when we want to explain the high diversity that we find in the Cape Floristic region. Okay, with that, thank you very much. I had an acknowledgement slide here, but they cut it out. Well, I have some collaborators and a lot of funding agencies that were involved in this, so thank you.
Thank you, Kerstin. Question from Adam.
Hi, thanks. Interesting, a question about the oxygen isotopes. In the modern record that we see in rainfall, it's very little variability compared to what we see in some of the areas that you showed where the other speleothemes came from. So my question is, to what extent do you think the lack of variability in the oxygen isotopes might be due to the fact that we have this sort of homogeneous ocean near us in the south, and less about the sort of amounts and intensity and seasonality of rainfall?
So we did a study, well, I mean, you're talking more about the winter rainfall areas here, right? So, I mean, we did a study of rainfall monitoring in Mossel Bay, and we did find there was a seasonal change of about 5 per mil in the rainfall there. So, you know, once you get out of the areas that are influenced solely by winter rainfall, I think you do get quite a bit of variability there. But I don't know if I would expect, you know, a place like California to have a much larger variability in the rainfall isotopes on a seasonal basis than it would be here. Like if the ocean is the driving factor, I don't know. I mean, over there, I could understand it more in the Mediterranean where conditions are a little bit more restricted, maybe. But I think what's more important here is really the, really huge changes in glaciation that we have on the northern hemisphere than compared to what we see on the southern hemisphere. Or I don't know if that really answers your question.
Yeah, thanks a lot. Actually, following up on that, can you also compare your estimates of climate stability to the tropics? Because right away you set it up, you noted that the Cape is actually up there in the very top of the tropical regions when it comes to plant diversity.
Yeah, so when we started out with this project, which was a few years ago by now, I actually did that. And all the tropical regions that I had records from had lower dispersion than the Robertson record did. I think we kind of moved away from that to simplify the, the whole publication a little bit. But for all that I can tell, I think the result would just be that it's lower dispersion and lower amplitudes there.
Okay, so in a sense, the climate stability may explain why there's a higher diversity here than in the northern hemisphere, Mediterranean regions, but not necessarily why it's up there with the most diverse tropical regions. Is that?
No, I think the other way around. Okay. Like all the tropical regions had higher dispersion than the. Sorry, did I say that the wrong way around?
Perhaps I understood it wrong.
Yeah, so Robertson had a lower dispersion than the tropical regions, or lower amplitude for sure. And I think, I think what happened at that point was we were like, okay, so our record is more stable than the tropics, but that's exactly, that was kind of the whole premise of the of the study. So we kind of threw that out to make it a little less complicated.
We can take one last quick question. I think, Fernando, you had a question?
Thank you, Kirsten. I don't know if I got that part, but the sites in the Mediterranean, did you get them from the literature or?
Yeah.
Okay, because I think it could be great because the one in the Western Mediterranean in Portugal is, to me, is very high latitude. It's close to the Euro-Siberian. I mean, and I think if you could go a place close to the Strait of Gibraltar, I'm sure in Goran's Cave in Gibraltar, they are in the very rock. You can get the speleothems there, and because I think the southwestern part of the Mediterranean is another kettle of fish.
So, I mean, for this study, I did several literature searches, and there's also a kind of global speleothem database project that I'm on the steering committee of. So, we've been working for the last 5 years or so to try to compile as many paleoclimate speleothem records, especially the stable isotopes, as we can. And I looked through that database multiple times. And, this is really everything that I could find that covers full glacial, interglacial stages. I think the research in Gibraltar, there's a big group working there from the UK. They've been doing a lot of monitoring, but I think they haven't really done a lot of paleoclimate proxy analysis there. So there's definitely big caves there, but I think they just don't have the records that I need. It's the same in Morocco. There's a bunch of caves in Morocco, but they all, most of them are Holocene or, just the last 1,000 years or so.
Thank you.
All right, thank you very much. I think we've unfortunately run out of time for questions, but yeah, can we just give Kirsten another round of applause? Thank you very much, Kirsten, for that talk. And then I'll hand over to Martina for some announcements.
Thank you, Kirsten. That was a really lovely talk. Just a few quick announcements before we close. We're going to have tea break now, a short one, and then the Global Forest Communicators session is on Hall 2, so on that side, and the symposium will be in Hall 1, as indicated on the program. The Turning Flowers pictures, many people were asking where can you get that afterwards. That was at the Tuesday evening session, those pretty flowers that they put over your faces. Apparently it's going to be available on the website or the app in about 2 weeks' time. Website, okay, on the website, thank you, Rupert. And then you can return your accommodation cards with Madeleine if you wanted to. Just make sure that she knows which room number you're actually returning. And then just a reminder, everyone had to be out of your rooms or their rooms by 10 a.m. So if you haven't got your stuff with you, you should please go and get it. I think that's all for now. Yeah, if there's anyone lifts needed or able to provide, please just look at the registration lists with Madeleine at the foyer or the conference foyer. And then we wanted to do a group photo with all you guys that are still left on day 5. So end of tea, we're going to shout at you and just ask you to move to the deck there so that they can take a photo of us. Thank you so much.
Before you run off, sorry. Yesterday, somebody lost a silver bangle in one of our late sessions. So I do have it. If you're looking for it, please just come and grab it from me. Thanks.
Okay, thanks everyone.
When we take steps to restore a forest, we play a part in something much bigger. We're making a better world for our health and for the health of future generations. By replanting and managing our forests sustainably, we create new spaces where plants and animals can thrive. We promote economic activity that brings work and improves lives. We make a real impact on climate change. We improve the quality of the air we breathe, the food we eat, and water we drink. We create a healthy environment for our children to grow. It's never too late to take action. Let's restore our forests and create a better future.
Some people don't care about me because they don't see my beauty. Some people don't care about me because they think I have nothing to give. But 2 billion people do. To them, I am everything. Their home, their food, their livelihood. But day by day, I disappear. And so does everything I give. 2 billion people care about me, need me. And now, I need you.
When we take steps to restore a forest, we play a part in something much bigger. We're making a better world for our health and for the health of future generations. By replanting and managing our forests sustainably, we create new spaces where plants and animals can thrive. We promote economic activity that brings work and improves lives. We make a real impact on climate change. We improve the quality of the air we breathe, the food we eat, and water we drink. We create a healthy environment for our children to grow. It's never too late to take action. Let's restore our forests and create a better future.
Testing. Hello, everybody. I think we should get started since we're running a little late. My name's Curtis Marion. I'm going to start off the symposium, and my talk is going to be a little bit of an introduction to what all the talks are focusing on. So it's my job to give you a background on what we're going to be talking about. Hello? Is it? Okay. Okay. Thank you. And then I'm going to give the final talk, which is kind of a synthetic talk that tries to pull quite a bit of it together. So what I'm going to do is I'm going to talk about the plains, the submerged plains, one of which we call the Paleogallus plain, and that's been our focus of research for about 10 years. So I'm going to ask the question, what are the submerged plains of interest? Why are they significant to folks like you and folks like me? I'm a paleoanthropologist, a person who studies human evolution. I'll give a very brief history of the research, what we accomplished as a research group, and then suggestions for the future. How do we move that research agenda forward, and what should be the research questions pushing that forward? So what are the submerged plains of interest? You can see the current coastline here, and this is the extent of the offshore submerged platform. And we've named these plains the Paleogallus plain, which is the one to the south, and that's the biggest one. And the smaller one off the west coast is the Paleobenguela plain. And we think that they were two separate ecosystems. They would be, of course, have some exchange, but we think for the most part that they're separate, and we've tackled them as separate ecosystems to reconstruct. So why are they significant? They adjoin some of the most important early modern human sites anywhere in the world. And if any of you went on our field trip yesterday, I talked a little bit, just impromptu, not yesterday, but the day before on Wednesday, about some of the South African sites. And I'm going to talk in more detail about those sites in my talk at the end of the conference. So these are some of those very famous early modern human origin sites: Colossi's River, Nelson Bay Cave, Knysna, which is a new one, Pinnacle Point, where I've been the director of the research project for 22 years, and Blombos Cave on the Paleobenguela plain. And then, of course, there's Deep Cliff Rock Shelter and De Kelder's Cave that adjoin the other ones. So that's the human origin significance. Why would they be important to you? Well, you often see plots that look something like this, and on the x-axis you often have area, because we know from many, many studies that diversity often correlates to area. And you can see the Cape up there on the upper left-hand side, and geophyte species diversity. But what's important for people who are interested in floral diversity is, as these plains are exposed and submerged over time at interglacial and glacial cycles, the position of that dot will change on that x-axis. And it was actually this slide in a conversation I had with Richard Cowling about this slide that stimulated the origins of our project, and I think got his interest a little bit into the project. And also, in the Holocene, of course, if you go around the Cape today, you see all these small buck, Chrysebuck and Diker and so on, dominating the ecosystems here. But if you look at the fossil assemblages in the Pleistocene, you had these big grazing species like zebra and wildebeest and some variety of extinct species. And it's always been a puzzle, where did those animals live? Certainly the current Cape flora wouldn't support them. But they're present, and they go extinct around 12,000 years ago. So these submerged plains are significant to two major research endeavors. One is understanding the plant and animal diversity and distribution patterns in the greater Cape floristic region, and they're significant to human origins. So very brief history of research. I don't have a lot of time. But really, the first publication that we all should have paid attention to was in 1972 by Dingle and Rogers. And I'm just going to draw your attention to that quote. Generally speaking, however, they would have probably produced clay soil, which would have supported a rich flora and fauna under suitable drainage conditions. So all the way back into 1972, these scientists recognized that these places would have been very, very productive ecosystems. And then Van Andel published a paper in 1989 Journal of Field Archaeology, where he took all of the topographic data that was available from the South African Navy and so on, and he digitized it and did reconstructions of the size of that plain. And this is the extent of the plain during isotope stage 4, which is at about 70,000 years ago. And then again, an important quote from him: "Unfortunately, very little information exists about the vegetation history, and for the glacial climate, the situation is hardly better." So he laid out a challenge for us in 1989. So despite loud and clear statements of significance from these prior researchers, archaeologists and ecologists pretty much ignored the presence of the plains. So if you go through the literature, in the archaeological literature, there's hardly a mention of the significance of the plains, even though all those major sites sit there looking out on what would have been a plain at that time. And also you see it pretty much ignored in the flora literature as well. So we're both guilty. In 2010, our research group published 2 papers. One of them was a speleothem reconstruction of what the vegetation would look like on those plains. And of course, Kirsten Braun just gave that wonderful talk at the beginning explaining how the speleothems work. And then second, there was a paper by Eric Fisher, where we did a structural reconstruction of what those plains would have looked like under different glacial and interglacial conditions. And those really were our first formal attempts to start to reconstruct what the plains looked like. And we came up with a basic informal model that kind of looks like this. So during interglacial conditions, the coast would be nearby. We'd have more winter rain on the plain, and the area would have been dominated by C3 grasses and more western flora. C3 vegetation in general, but the grasses would have been C3 grasses. And then during glacial phases, you would have had a plain that sat here, the coast would be far away. We would get more summer rain, and this was signaled by the speleothems that we had. And those C3 grasses would have been replaced by C4 grasses, and the more eastern flora would have swept into areas like Mossel Bay. In 2011, John Compton published a paper in Quaternary Science Reviews where he did a basic vegetation reconstruction, largely based on what the geology would have looked like, his reconstruction of the geology of the plains. And then in 2012, we got together at a workshop in Cape Town, which was designed to, in a sense, create an update from the classic book that Richard Cowling had published. And in 2014, that publication came out. And we wrote a paper that summarized a basic general model for what the plains would have looked like. And then in 2015, we published 2 papers. One was senior authored by Janet Franklin. We wish she could have been here, but she can't be. And we laid out a research agenda for trying to reconstruct the paleoecology of the plains. So that strategy advocated switching from a site-based inductive approach, where we take individual records and try to infer, to a more deductive model-based approach, where we would use climate simulations to try to project the vegetation. And those would be, we would build paleoscape models that are then tested with these empirical records that people like Kirsten would generate. So our workflow is we would start with regional climate models and structural models, right? Reconstructions of the soil and geology, and these are computer-generated, supercomputer-generated climate models. And those would drive vegetation models that our vegetation experts would generate, and they've done that. And those vegetation models would then drive faunal models, where we would reconstruct what the fauna would look like on the plains. And then using experimental work of the type that Jan de Fanck and Suzanne Botte will talk about later, these are foraging experiments. We would build what we call resource-scapes that would explain or predict what the habitats would be valuable, how valuable they would be to foragers. And then build human behavioral cultural models. So we would actually create agents in the computer that would be hunter-gatherers that would forage these landscapes. And overall, that creates a paleoscape model. And then, of course, a paleoscape model is organic. We could feed into it data like hypothesized changes in fire regimes to see how they would affect the vegetation. So in 2020, we published a special issue in Quaternary Science Reviews on the Paleoegolist plain. It had 22 papers that covered all aspects of that workflow that I just explained to you, and it sketches a general model under last glacial maximum conditions. So last glacial maximum would be the maximum glacial, which is really between around 20 and 18,000 years ago. And that's what our model in vegetation reconstruction looks like. And there are 20, 30 people contribute to this model. And I'm not going to go over the details, but one of the things that's really significant about it is it projects a savanna ecosystem right off the coast in front of those very important origin to modern human sites that I pointed to. And we never had any idea that that ecosystem would have been there. And that's where those grazers are and those extinct species and so on. And what you'll hear about in this symposium is the details of how reconstructed that model from the people who actually did all the work. And of course, I'm one of the project directors along with Richard Cowling. Richard really developed the vegetation model, and I do the archaeology, and I kind of herd the cats. So that's my job. But I want to point out a classic statement by the famous statistician George Box: all models are wrong, some are useful. And I want to point out that there's almost certainly there's aspects, specifics of this model that are wrong, and as going forward, we need to improve its quality. I'm hoping that we can excite some of you to be involved and help with your expertise in flora and fauna to improve that model. But I do want to assert that this is a very useful model going forward. So what are our next steps? What we've done is we've created a model at glacial maximum conditions. And it's a really good model for the last glacial maximum. And it's a basic model for MIS6, which is also a major glacial phase. And of course, model 1 is the current conditions, right? That's what we have now. This is a sea level curve on this axis and age here. And you can see sea level has changed radically. So model 1, the current conditions sit here. Model 2 sits there. We haven't yet approached that model. Model 4 would sit there, which is a glacial phase that's kind of in between the maximum glaciers and the current conditions. Model 4 is the one that we've created, of course. And I'm going to argue that the one we really need to hammer next is isotope stage 4. And the reasons for that I'll talk a little bit later at the end of this symposium. We need to expand to the Klein-Karoo, and we need to expand to the Paleo-Benguela plain. And thank you very much for your attention. Yeah.
Thanks very much. You put up a figure showing area and
geophyte species diversity.
Species diversity on the Y.
Yeah.
And you showed a shift to the right in terms of area. And it got me thinking, wouldn't you also, or would you expect a similar shift in the y-axis?
You would right, because area, well, a portion of diversity is explained by area, right? The classic species area curve theory developed by Rosenzweig. And over the years, whenever I've looked at those graphs, I said, yeah, but the x-axis is always in flux over time, right? And so when we do these species area curves, we're taking modern diversity and plotting it against area. But of course, area changes over time, and we live now, and that's just the end product of area. So I think, there might be a way for the paleoscientists and the modern ecologists to come together on this, and we try to take that x-axis and make it more organic to reflect the landscape histories that we're really interested in, that produce that diversity. You see what I'm saying? So to me, that's an incredible area of collaboration between people like me who work in the paleosciences and people who dominate this conference. And that's part of the reason we're giving talks here, is to get people interested in the paleo side and so on. Yeah.
No, thanks. That's great. Thanks.
Just a quick question. You mentioned in the last glacial maximum there was an increase in summer rainfall in the east and an invasion by C4 grasses, but was the temperature suitable for C4 grasses?
Well, okay, so we have very poor proxies for temperature from paleo records, and it's not from lack of trying, but it's just a really hard number to reconstruct from paleo proxies. We have done a last glacial maximum climate model. So we've taken South Africa is very fortunate in having a high performance climate model, right, that has been developed by your scientists. And you know, you're using it for projection of weather and climate in the future. And we convinced those folk to collaborate with us and take that model and run it at glacial maximum conditions. Okay, we don't have a talk on that, but there's a publication on it. And so we split the time on USA supercomputers and South African supercomputers. And it's a very powerful model. You know, you're very fortunate. South Africa is very fortunate in having its own high-performance climate model. It shows us that last glacial maximum conditions weren't all that much colder here during glacial maxima. Now, if you were up in France, temperatures during glacial maxima would probably be 15 centigrade colder, but here it's probably more in the range of 2 to 3 degrees centigrade. Okay?
Thank you.
Yeah. Okay. Okay, thank you very much.
Hi, good morning. My name is Hayley, and I'm a marine geologist who works on this project too. I'm going to ask you to imagine a landscape under the sea, would pretty much wrap this up. And I'm going to give you two very quick lines of context first, which I think are important to just sort of mention up front. The first is that in marine geology and geophysics, it's actually a very new field, and we haven't been able to map the seafloor since the years succeeding World War II. So only for the last 70 years, there's actually been technology to image the sea floor by using methods of sonar and magnetics and other techniques that were initially developed during the war. Because there are no remote sensing methods that can look from space through the seawater and map the seabed for you, it stands at the moment that the planet's a better map than our own seafloor. And it's a little bit technically challenging, and as such, we have to mount instruments on boats or ROVs or AUVs or any transport vehicle and steam up and down, and we call it mowing the lawn, to collect these kinds of data. So there's no remote method of collecting seafloor data just yet. The other line of context is just to say that sea level is certainly not static and it fluctuates continuously. Obviously in geology, we're always thinking about deep time and processes from ancient time past. I'll just keep talking. And in the last 500,000 years or so, since the world has been in the current climate cycle that we're in now and has been familiar for the last couple 100,000 years, sea level has fluctuated from a maximum high stand of about 10 meters above present to a minimum low stand of about 130 meters below present in these approximately 100,000 year cycles. So the implication is that for most of the last 500,000 years, the part under the red line on this little curve has been exposed as a terrestrial landscape. So for 90% of the last 500,000 years, the seafloor was not under the sea. And we're able to now use these fancy methods to map this area and to reconstruct these environments. Obviously, as sea level fluctuates, it takes with it beaches and dunes and wetlands and rivers and all the features that you find along a coast today. So the implication of that is basically that this notion of an Atlantis is actually everywhere, all over the world, all over the continental shelves, and they're exposed every 100,000 years or so. The real impact of these places, though, is of course where human populations are involved, and ancient human populations. And that's what makes our work on the Cape South Coast and in the Cape Floristic region quite special. Zooming in onto Mossel Bay and the Cape South Coast, as Curtis has just mentioned, these very important Middle Stone Age sites sits adjacent to the Agulhas Bank, as we know it now, in marine geology, or next to the Paleo-Agulhas Plain when it's exposed as a landscape. And it's a very wide, flat topographic area. So when sea level is low, when sea level is 130 meters lower, there's about 100 kilometers more of space before you reach the coast off Mossel Bay. And this width, of course, varies depending on where you are, because the shelf is not a homogeneous width, as you well know. So our work aimed to try and understand what's below the water. And specifically, my and my colleagues' work within that was to treat the shelf and the coastal plain as one continuous feature that's periodically exposed and periodically drowned. We realized from the much earlier work that Curtis mentioned of Dingle and Rogers and Van Andel and John Compton, that this is a significant part of the landscape to try to reconstruct. And with the methods that we have available now, we were able to do that. Then, of course, once the geology is reconstructed, link it into human populations. And you'll see more on that in the talks that will follow. So we did high-resolution surveys around Mossel Bay and Fleas Bay at first, where we had full coverage of the seafloor. And then onward from that, in the next 8 years or so that followed, we collected as much regional data as we could to try and get a broad picture of this underwater landscape. So my colleagues and I spent 3 months working in Mossel Bay, collecting side-scan sonar, multi-beam bathymetry, and 2 types of sub-bottom profiles across this part of the seafloor. We then went diving. We're also commercial divers, and we dived between 12 and about 50 meters, approximately 50 times, and we collected rock samples that we could have dated using luminescence methods that reveal when last these beaches and dunes were actually beaches and dunes. And a grain of sand from the middle of a really big sample can tell you when it last saw raw sunlight, which is pretty amazing. And that's the best method to date these rocks at the moment. Of course, Mossel Bay is not known for its tropical diving, but rather other stuff. And we had to take some necessary precautions. But these are the kinds of samples that we hammer off and send up from the seafloor using lift bags and things like that. The visibility is not great. We usually had about 20 centimeters or less on the bottom, but we managed to get quite a lot of good material. Following these sort of investigations around Mossel Bay, we had a bilateral project that started, a German-South African collaborative project that allowed us use of the Meteor vessel shown up there in the top right. And we were able to take sediment cores through all of these features that we'd already mapped with geophysics. So I knew where there were paleo river channels, paleo lagoons, all sorts of interesting environments on the seafloor that were holding terrestrial deposits. And we were able to poke holes into those and take samples of them to get a glimpse of the actual sediments from those kinds of deposits, right back to the last glacial maximum. So in all, we started broadening it outwards. I was able to look from Cape Infanta to Plettenberg Bay, and then, as I'll show later, incorporating legacy data to get a kind of a picture of this Paleo-Agulhas plain at different time periods. We're all familiar with the shape of the coast as it is now, but as I mentioned earlier, these things are totally, they're totally variable. The sea level changes all the time. Coasts change all the time. The seafloor is full of beautiful features that can be imaged with sonar. And I mean, just for example, the Cape Canyon right next to us here at Langebaan is actually bigger than the Grand Canyon. And a lot of people just don't know that. It goes down to depths of 3,600 meters. So stripping away the water to a depth of minus 130 meters, which was the time of the last glacial maximum, or any glacial maxima, here we get a real picture of this complete landscape. This is more like the complete Cape Floristic region, as we should be imagining it. A little bit quickly about the geology. So Anton gave a beautiful overview of the geology on the first day. And pretty much we've got Malmesbury Group Neoproterozoic deposits, which are brown, Cape Granite, which is pink, the Cape Folded Belt, which is blue and green, and then Mesozoic rocks, which were laid down when the oceans opened, which are shown kind of in an orange, and then coastal Cenozoic deposits in yellow. When we strip away the water, we see that there's a dominance of the Mesozoic and the Cenozoic rocks and sediments under the sea. So the Cape Fault Belt doesn't extend very far out to sea, except for here around Cape Agulhas, Cape Town. But on the Cape South Coast, you have onlapping sediments right next to the coast that are siltstones and claystones and very different things to what we have now on that heavily dissected coastal plain. So everything we're doing under the water, you've got to kind of switch gear in your mind and forget what you know about that current coast. Try to imagine something that looks very different. This is a really good, Southern Africa is a good place to study sea level changes in these kinds of deposits because we're pretty tectonically stable at the moment. And we're very far away from the ice sheets. So we never actually had ice in the glacials, and our deposits have never been scraped off by moving ice sheets, for instance. Here's a picture taken on a very rare, clear day from Cape St. Blaise, where the swells were not crashing against the waves. And you can see that these rocks terminate almost immediately. Of course, there's marine sand out here that buries the Mesozoic siltstones and claystones, but they're just there, just next door to these cliffs. They do extend further out to the east off Cape St. Blaise, but generally they end right next to the coastline. I mentioned earlier that we integrated legacy data in making these maps, and it's always, of course, important to acknowledge the work that's been done before and to be able to draw upon what they learned from it. So they are early on bathymetric and sediment maps that were very regional scale, of course, and we could integrate that with our higher resolution products. So looking at some geomorphic features that are on the seafloor, we found cliffs and caves. This is off Cape St. Blaise, so those things extend out, exactly the same as what you find on land now around Pinnacle Point and along Cape St. Blaise coast, except that they're under the water. We did dive in some of those caves. We never expected to find archaeology, though, because we've got a very high energy coast. As sea level was rising, it probably chowed out most of those soft deposits. And even nowadays, Curtis will tell you that when they're looking for good archaeological sites, you've got to look higher than about 8 meters above present sea level to be able to get things that are quite undisturbed. So we never aimed to find archaeology under the water. We were aiming to reconstruct the environment that was there when people were using it. Some other features that we found were off the Great Brack River. This is where we did all of our diving. They're beautiful paleo coastlines, cemented rocks and cemented beaches and dunes into rocks, Pleistocene rocks, extending out there in this sort of step progression, and they've been cut by the Great Brack River, so we knew they were quite old before we even dated them. For the most part, though, it's quite a sort of flat shelf, but it's definitely scattered with these Pleistocene rocks. From the regional sub-bottom profiling data, we were able to find cores of those cemented dune rocks and figure out that there were really big dune fields out there during times when the sea level was low, shown in dark orange. Those are where we predict there were dune fields, probably never got very high because they had a lot of accommodation space to spread, and sediments will always take the path of least resistance. So dunes won't get high if they don't have to get high. And, oh, that sounded terrible. So there are quite laterally extensive dune environments out there on the shelf, probably pointing towards things like Strandveld. One interesting thing that I just wanted to mention is that we found a snapshot of time from coming out of a very harsh glacial period 160,000 years ago. Sea level was rising up towards the next period of warming. Early humans were using this environment. We found a time when sea level was rising very quickly, probably 4 or 5 meters per century, which is 4 or 5 times faster than now. And we see this beach dune package that overstepped in response to a fast rising sea level on a very flat shelf. And the implication of that is that people would have probably witnessed the loss of their landscape within a lifetime, which is quite profound, and it's quite different to the scenario that we imagine now, but possibly something we could look forward to in the coming years. So just to say that these things have happened in the past as well, from purely natural cycles and processes. So to make a map of the Paleo-Agulhas Plain, I had about 40 or so bits of data, different resolutions, different types of data sets. And I thought, how on earth are we going to pull this together? It's very difficult to make maps for things on different scales and from different areas. So I resorted to dusting off our old light table at the Council for Geoscience, printing things out to scale, using tracing paper and literally drawing a map. Of course, based on scientific validity. It wasn't a sort of nonsensical map, but pulling together all the bits of information and starting to extrapolate between them. So it's also good to borrow from technologies that are several decades old. Ultimately, we pulled together all of this data from cores, from samples, from geophysics, from old data sets, from grab samples from industry, anything we could find, and produced a map of the last glacial maximum, which is 20,000 years ago, of the Paleogalus Plain. It's the first time we know of that anyone's made a geological map of the last glacial maximum, so we were quite stoked with our map, and we were able to use this base data set for lots of different things. I want to point out a few profound differences. So firstly, you can see the dominance of the different kinds of colors on the seabed compared to what's on shore. We knew that already. But what we didn't know is how the rivers behaved on this flat landscape until we were able to image them with sub-bottom profiles. We see that they're very shallowly incised and they spread out across very wide distances. The Gouritz River, plus its floodplain, gets across 18 kilometers. It just blew my mind when we saw this from our data sets. These rivers would have obviously promoted different kinds of environments out there, and it even seems that the Gouritz splays into a delta at the time of the last glacial maximum, and that's based on a sub-bottom profile with multiple channels over the same kind of area. There's a volcanic feature out there called the Alfred Banks, and people that dive will probably know about it, or fish maybe, which produces very different geologies and soils. And there are a lot of dune fields, coastal dune fields, from the time that they were coasts, shown in yellow. From the geology, we were also able to infer something about soils, because we cored through some of these soils with the German ship. And there are various types of hematite minerals, for example. And these things have accumulated, of course, with the estuarine deposits, the lacustrine environments. And from these sort of snapshots from the sediment cores, and from working closely with Richard Cowling, who knows a lot about soils in the Cape Floristic region, we were able to take the geologies that we had, some snapshot samples, and start to infer something about what kind of soils we might expect. Like Curtis said too, this is a model, it's a conceptual idea. The more we can sample it would be the better, but yeah, it's just something for us to base our further interpretations on for now. Going through quickly some highlights of other things we've learned from mapping this offshore area. One of them is that vegetation constructions were enabled through the geological and soil mapping process. We knew that we were going to expect a different kind of environment, and we knew this from the geology that was already mapped. We knew it from the archaeological records in coastal caves that had a lot of grazing animals, for example, that just didn't make sense. And now we've been able to demonstrate that too, from the actual kinds of deposits that are out there. Working with Charles Helm, who's a person on our team who studies ancient trackways, we have a little sliver of the Paleogalus Plain still cropping out along the coast in the form of these aeolianites and things, these cemented rocks. And through mapping out these animal and human trackways, it gives another perspective on this paleoecology. So these areas along the fringe of the coast now are probably examples of a much bigger system that's now under the water. There was a lot of water out there. There were lakes. For example, off the Great Brack, there's a lake the size of Langflade Wilderness. Bear in mind, though, that a lot of these big rivers would have probably been estuaries because of that flat landscape. So a lot of the water may have been brackish and not drinkable. So we're also looking at the presence of possible stromatolites on the continental shelf, because stromatolites, we have them along our coast, especially along the Eastern Cape coasts, and we see that they actually pool fresh water along these salty coasts. So these we think would have been important resources to early humans too. Beach shape and coastline types changed radically through time. As soon as you get below a depth of 47 meters, which is very specific, but that's the magic number, you no longer get these embayments along the Cape South coast. You would have had fairly straight sandy beaches because we lose the Cape Fold Mountains. We lose that ability to make those embayed coasts. this has an impact on, of course, what kind of shellfish are existing in the area and what people had access to. And thankfully, we were able to compare the sort of broad-scale geophysics data to very high-resolution cave data and test in both directions. And we find that there are times when the record is dominated by sand mussels, times that it's dominated by rocky coastal species, and times when you have both. And the geology also sort of talks to that. We can strip off these marine sediments and see what lies beneath from sub-bottom profiles. So we think that the only little analog we have of this Paleogalus plain, of our 40,000 square kilometers underwater in this part of the Cape Floristic region, is the Agulhas plain, which is just south of Bredasdorp towards Cape Agulhas. We think that that's a little portion of it that still sticks out now, and is possibly the only suitable analog that we have at the moment. Then wrapping up, I just want to sort of say that to be able to look into the past enables us to also consider future scenarios. We know that sea level rise, for example, is a certain consequence of global warming. How did people deal with stuff like this in the past? How can we sort of keep that in mind for our future-looking plans as well? So a lot of what we probably should keep in mind for general understandings of areas and plans, even looking forward, we should always remember the past scenarios that are sometimes pretty different to the ones that we know now. I think I'll leave it there. Thank you.
Thank you. Fascinating stuff. Just here's a curiosity of a botanist who knows nothing about geology. Do you have any reason to expect that this paleo landscape extended right round into KwaZulu-Natal? I'm asking out of curiosity of linkages of plants in the Agulhas Plain and the area around Umtavuna, which also has got sandstone, and wonder whether there could have been a connectivity and dispersal possibility. Thank you.
Sure. So that's a great question. So the shape of the continental shelf around South Africa is very variable, and I didn't show a map of the whole coastline. But from Port Elizabeth, Rebecca, it narrows very much. And there's a very skinny, steep continental shelf, all the way from Cape Padrone to Maputoland. And it sort of opens up again in Mozambique. So the whole eastern margin has a very narrow shelf, mostly less than 10 kilometers width, which is pretty significantly different to what's down here. It's also underlain by quite a lot of bedrock, especially around the Wild Coast area. So I would imagine that might form some form of a barrier, maybe to species types that perhaps came later or took hold when the sea levels were low on those continental shelves. I'm working on a project in the Wild Coast area as well, where we're doing a similar thing, and we're finding stark differences. And we're finding archaeological sites with last glacial maximum deposits right at the coast. So people never left those specific sites because they never had to, because the coast was never too far away. So it makes for great comparisons between areas with wide shelves, like in the Cape Floristic region, and these areas with narrow shelves, like up there in the East Coast area. I come from the KZN South Coast too, so I know that quite well.
Thanks, Haley. That's so interesting. How do you respond to these, the mining and the drilling and so on offshore? I just think it might sort of mess it all up.
Yeah, I mean, we don't have, okay. I suppose drilling and mining would be two different things. So I suppose that's oil and gas and then minerals that you're referring to. Where I work, we're a research council, and we don't really have any involvement in any of those sorts of activities. But of course, we look at the data that are collected. We've done some models on, like, in the hydrocarbon sense, if they don't cap the wells well enough, where will those hydrocarbons float to, looking at that kind of thing. We've done a fair amount of peripheral work around that. Certainly the most destructive things that you could probably do on the seabed would be to dredge the seafloor, because then you're unselectively taking absolutely everything, scraping that layer off the top. And there's extremely high biodiversity, especially in mineral deposits. Like, you'll have different chojos living on manganese nodules to right next door to them on the seabed itself. And that's very poorly understood, actually. especially in the deeper seafloor environments. Every time a biological scientific research project works out there, they discover something new to science. So I think we have to keep that in mind as people are developing legislation around this, acknowledging the things we don't know in trying to build responsible processes that keep in mind that technology is going to change, and maybe trying to force technology to change. So saying, like, if no dredging could be allowed, then you could come up with something else that's not going to be as destructive. What freaks me out is the way that the coast has been reclaimed and modified along the Northern Cape coast. Richard took some of us to Namaqualand in June. I was quite horrified at the mess that the diamond industry has made and left behind. And I know that they reclaim the coast as they try and mine outwards instead of working under the water, rather just make it subaerially exposed by dumping sediment there. And you look at this place that's been shaped by very long-term processes, and within a few days a human being can mess it up, and I think that's quite terrifying. So I haven't done any specific work on that, but I do take part in some of these processes towards policy development and legislation development. And I'm working on a UN project in the deep seabed, and it's interesting to see that nowadays we can bring sustainability to the fore. People do listen better than I think they did in the past. And unfortunately, there are these examples to show of what damage can really be done. So, a bit waffly, but it worries me. Yeah.
Thanks, everyone. I'm just concerned that we're running a little bit behind time, so I'm going to suggest that we stop questions there.
Sorry.
If you do have any other questions for Hayley, please put them on the app. And I'm sure Hayley is happy to chat to you during tea time. So our next speaker is Johanna van Holt. Thank you, Hayley.
Hi, thanks. Thanks, Adrian. I haven't been called Johanna in a while. Living in Cape Town, but I'm from Pretoria, so I guess it is Johanna, or Johanna. So I form part of a team studying spatial patterning in the landscape, commonly known in the US as mima mounds and locally known as yivulkies, if you translate that directly, for those of you who are not familiar with it, called small hills. So what I will present today... Oh, you can't hear me. Can I stand closer?
This will give you more flexibility.
Oh, yeah. Okay, no, I'll use this one. Okay, sorry about that. So what I'd like to show you today is, obviously, Mike and Jeremy has been part of this team for quite a few years, and then I came along. And so what I would like to show to you today is to make a case for the importance of these yivulkies in terms of how we understand the landscape in the past and in the present and potentially also in the future. We've recently also have some collaborators from Nanjing doing some dating for us. And so this site is in Inverdoorn to the northeast of Cape Town, some of the most beautiful yivulkies around, the circles with some euphorbia species around the outside of the ring. So spatial patterning occurs all over the world and comes in various forms, from linear to gaps to labyrinth forms to spots. And in southern Africa, we have a huge variety of different forms as well, from fairy circles to Nepka-like features. But today, of course, I'm talking about yivulkies. And within the yivulkies itself, we do find a large variety of different forms as well. So it's not just one particular single type of patterning. From the patterns that we see on steeper slopes in the Cape Fold Belt, fairly well-developed patterns, to more sort of emerging patterns, and then here to the at the bottom right, all the way to the east, close to the Great Fish River, what we refer to as bush clumps. And sorry, my scale dropped off. I don't know what happened to it. So those bush clumps are also about 30 meters in diameter, and they consist of sort of more small tree species on those patterns. But then we get obviously the more sort of archetypal yivulki that you often see in images, and they vary considerably as well, from the Inverdoorn Let me just find the laser. From the Inverdoorn site to Kapelsfontein, which is just a few, sort of 50 kilometers to the north of Inverdoorn. Very flat, hardly any mounds, bare spaces, to the sort of the stuff that we see all along the west coast, and some of it buried by rubified sand, as John Compton likes to call it. And these Yvelkes vary from significant to marginal regular spatial distribution. So we'd like to think that this is as a result of water-driven processes. So after this diagram from Aron, basically how the vegetation shapes this overland flow and the infiltration, interplay between soil crusting and the vegetation, how it changes that infiltration. And then interestingly, so obviously, well, not obviously, but Ferry circles more into this sort of soil diffusion model. And then this water conduction, which actually I thought about when we were in the field trip with Richard Cowling mentioning the, what is the plant species with the? Sergia. Sorry, disclaimer, I'm not a botanist and I'm not a paleoscientist, so yeah. But anyway, so this is what the mechanism that we think is responsible for this. And so we hypothesized that Yvelkes occur in relatively arid areas. You need the sort of the scarcity of water linked to cool season precipitation, subject to low evaporation rates, that creates conditions for vegetation patch formation and erosion. Our predictions are that they occur predominantly in areas where rainfall is concentrated in the winter months. The long-term average rainfall is low, so that the vegetation cover is constrained, so that vegetation patches can form. Dry summer conditions that limit vegetation also promote aeolian deposition and erosion, and that's how I was brought in as part of this team as a dust scientist, testing the aeolian deposition sort of hypothesis, and we tested it in one of the previous papers that we published, that the actual pattern is not set up by aeolian processes. Cannot form in fire-prone landscapes, since fire resets the vegetation and would remove long-term pattern formation, and paleoclimatic factors combined with changes in coastlines might have determined current distribution. That is a little bit of a sort of a long shot that we've got going there. But anyway, let's get into it. So what we did is, and this is through the years working with Jeremy and Mike, this has been done before. We've done it again now, actually extending the range of the Google images that we take from 10,000 random points, and then we manually screen these images. And then categorize it according to type of mound, really looking carefully at the years that we've spent in the field, looking at the different types and of these Yvelkes that we see. So I just want to make sure this timer on here, is this a? It's the whole session. Oh, okay. Oh, sorry. I was just getting worried. All right. So we categorize these mounds, we use the BRT, and we used a whole bunch of different variables, potential predictive variables, from climate, edaphic, vegetation, topographic, fire, everything. And from our classification, so we see Yvelkes in 8% of the images classified actually within the Cape Floristic region. So from this, also, through the years, many field trips spent looking at using drone imagery, soil characteristics, including particle size, the composition, the depth to hardpan, using a dynamic cone penetrometer to get a profile under the mounds. Dust characteristics, we've put out passive dust samplers, and then now giving it a stab at OSL dating. So I'd like you to just take note of these sites. So this is the distribution of the images that we've classified according to Yvelkes. And then Subatsfontein, there were 2 sites, and Maskam, those 3 sites are the important ones which I'd like you to remember for later on. So here, our drone imagery, it kind of, we'd like to think it shows that model by Maron, where the sort of vegetation intercepts the drainage, so the slope and flow direction going that way, almost the mounds capturing some of the water flow as it moves down the landscape. Our predictors, going back to the BRT, these are the strongest predictors, and we've classified them according to these main categories: the winter precipitation, productivity as kind of represented by NDVI, temperature, aridity water, and then sediment. And so I'll just go through a few of these and talk to some of these partial dependence plots from the BRT. So precipitation, so the proportion of winter precipitation, so that's from April to September, was the strongest predictor, and we see that there needs to be this sort of concentration of that rainfall in the winter. And then also aridity, of course, important, as we've said, for this pattern to form. NDVI, there is a particular range, fairly low, but not too low to be able for the pattern to also actually establish. And then the variability in the NDVI actually also, the seasonable variability also ended up being quite important. So these particle soil characteristics are always a little bit controversial, I guess. Sand and silt being correlated with each other, and then soils grids having its own issues. But from this, we kind of see that sediment characteristics is important and fairly low, surprisingly, fairly low salt content, and we see that from the Subatsfontein and Muscam yilkies that we've sampled and we've tested the particle size, that the salt actually is within that range. If we look at the samples that we've taken, and of course extremely sandy as well. Fire, there were only 12 sites, images, and the Google images were 640 by 640 meters wide, wide and high, I suppose, the dimensions of them. So only 12 of the 782 of the images were classified as containing yilkies, and only 12 of those at mound sites in locations that experienced fire in the last 20 years. This is the Modus area burn product, and so we kind of conclude that fire excludes yilkies as it would reset the pattern. The vegetation just it occurs across diverse vegetation types. Sorry, the colors are not so easy, but Strandveld, succulent karoo, summer Nosterveld, fynbos, and if we kind of look at where they are distributed, so this is the Yilkie site, so that's each of those photos per 100 square kilometers. Most common in the Strandveld in terms of density, and then succulent karoo, Renosterveld, kind of disaggregated that fynbos from those, and then azonal. So seemingly potentially saying that the geological substrate not a determinant, as Aeolian sands, some of the shale, others sandstone. So this is our predicted distribution. Not just a disclaimer that we didn't implement a threshold in the BRT here, but it follows the winter rainfall, but it extends further east, and so we would like to ask the question why. And we did a campaign to go and sample some soils, some actual cores in profiles of 3 Yilkies, 2 in Subatsfontein, as I pointed out, and 1 in Muscam, close to Van Reinsdorp. And we sent this to Nanjing in China for OSL dating. It took quite a while because of COVID, and China was locked down and nothing happened. And so some of the dates that we get in the Muscam, a very kind of more degraded site, in the massive calcrete, 22,000 years. And you might ask, because we were worried about bioturbation, that we would get any meaningful chronology on these samples. And so we were surprised to see that it actually does make sense. So they haven't dated everything because we wanted to just have a first stab at actually looking at whether these dates made any sense. So from the bigger Yvelkes in Subatsfontein, 25,000 years, and then the biggest one, a monster of a Yvelke, actually showing sort of 2 accumulations of calcrete, quite a bit older. So we need to figure out what this means, and some of these dates agree with previous work done by Midgley and then Potts et al. As well, who showed similar date ranges to these dates that we get. So we'd like to figure out, none of us are actually paleoscientists by training, so we'd like to figure out actually what is going on, and some recent work that is unpublished by a group that includes Brian Chase, looking at the inland paleolakes and the different lacustrine phases with some of the dates that they get, and it is quite interesting how these sort of line up with some of those dates. So, we'd like to explore this quite a bit further, but we obviously need some help in terms of the paleoscience behind this. And then could we potentially, so Curtis Marion was talking about, this is a paper from Francois Engelbrecht, Richard Carling and Curtis Marion was part of this paper, and he was referring to this paper, I think, earlier, where these climate models, these are different, eight different simulations, these regional climate downscaling, the models projections of the LGM. And so I just highlight 2 sort of extremes, this one and then this one. And we were starting to wonder, could we use Yvelkes, and are we selling them short? Could they be used as useful paleo proxies to reconstruct some of what has happened in the past? So that's kind of I guess a fresh idea. Previously the focus has very much been on what is the origin, and these different schools of thought about what that origin might be. But now, sort of, could they actually be much more useful than what we previously thought? So then, looking into the future, currently in the future, and funny how it's come full circle. And me, myself as a dust scientist, with a group of scientists from Basel, we've actually, as part of my PhD, I looked at remote sensing imagery to identify dust from the Namib Desert, and I did include the southwest coast of South Africa, and through the years, I've seen an uptick in dust from the west coast of South Africa. And the last 2 years have been extreme. So we see these massive plumes. So I think there's always been dust coming off the west coast of Southern Africa, but the plumes that we see from Landsat and from MODIS are massive. And they are mostly driven by human modifications. Whether you look at the QSEP and the diversion of the northern arm of that delta, the Omdal Dam that was built, we went up there, we went to go and put dust samplers out. Of course, they were filled within one event of the east wind. But talking to the community and the history of that area, the land use, coupled with drought, potentially fueling this massive emission of dust from this area. And what we're seeing is areas that are degraded down to the door bank, where nothing will grow anymore. Areas that are just sand accumulations covering the infrastructure. Many times the community, after the east wind blows, they actually can't can't get out of Eksteenfontein. So an issue, because this landscape is being degraded. As Frank Eckhart, who was my PhD supervisor, always used to say, uses the analogy as like a zipper. Once you start opening that up, there's nothing to stop it. So the community desperate for help, and again, not my area of expertise, but we're trying to work on some kind of way to intervene in saving this landscape. So that is just the area. I think it has emitted dust for a very long time, but really turning into the extreme, as you can see here, these last few years, quite hectic. And very much a story of, as I said, drought and overgrazing. And the community acknowledged this. So just as a conclusion, they're fun. They're certainly not my real bread and butter in terms of my career, but I've learned a lot from Jeremy and Mike. which I have to thank them for. I guess that's my thank you slide. So they're likely the consequence of water-driven processes, we think, structuring vegetation pattern. They provide important clues for landscape evolution and might be useful for reconstructing paleoclimates. Then 8% of the GCFR landscapes and contribute to landscape heterogeneity and species richness, which is why they're important to preserve. And then they're degraded by land use, including agriculture and mining, as Hayley's also alluded to, vast areas of that landscape. And that's it. Oh, and I didn't mention the termites. I'm sure there are people that are sitting there, but when is she getting to the termites? And I just want to mention that, we don't think that it's set off by termites, but of course bioturbation and faunal activities play a very important role in the feedback and sort of, perpetuation of these mounds. So I'll leave it there.
Thanks, Joanna. Thank you very much. Unfortunately, we don't have time for questions.
Sorry.
We're running quite a bit behind time. So we're going to move on to our next speaker, who is Adrian Grobler. So I'm going to go up there now. Jasper Spingsby, can I ask you, please, to do me a favor? Should I start clapping when you get to 10 minutes? Start clapping. Thank you. I'm a bit shorter than most people. Can you hear me? Is that fine? Okay. So I'm going to, this was supposed to be a long talk. I'm going to turn this into a lightning talk, just so that because there's a whole bunch of really interesting talks. I'm done. Thank you very much, Jasper. That I want you to see. So I'm gonna try and get through this, and then we'll move on to those other talks. So, yeah, what I'm talking about today is just a project that we're working on at the moment. We're trying to figure out basically the importance of C3 and C4 grasses and CAM shrubs in paleovegetation of the Greater Cape Floristic region, particularly during glacial times. And what we focused on was the last glacial maximum around 20,000 years ago or so. And yeah, so we used paleo distribution modeling to try and get an idea of what those spatial patterns looked like. And essentially what we found is there weren't any huge major upheavals in the distributions of these plant functional types. There were some market changes here and there, but mostly they were restricted to specific landscapes throughout the Greater Cape Floristic region. So, just to give you a little bit of background and to tell you how we got to that answer, yeah, South Africa is a very diverse country, as I'm sure you all have heard multiple times. Climatically, it's quite a complex zone. These are the Köppen-Geiger climate zones for South Africa, and as you can see, the Cape outlined there in black has quite a bunch of different colors there. So climatically, it's quite complex. These are based on basically rainfall amounts and temperature regimes, and we also have varying gradients of when our rainfall falls. So that red line just indicates sort of a not quite arbitrary, but a boundary between the strongly winter rainfall on the left-hand side and the more all-year rainfall zone on the right-hand side, where where rain can fall any time of the year. So these different climatic regimes basically just interact with each other, and they influence the distribution of these various photosynthetic plant types that we find, C3, C4, and CAM. And the reason we are interested in this is, as you've seen from Kirsten's talk and from Curtis's, I think Curtis also mentioned this, is that we can, we're interested in finding out about the carbon isotope ratios, because this gives us an idea of what environments would have looked like back in the past. We can use this to reconstruct climates, and we can use this to reconstruct paleo vegetation. And just to show you quickly here, This is the range of isotope ratios that we can find, and usually there's quite a distinct boundary between C3 and C4 plants. So that's how we can tell them apart. So just very quickly, what did we do? Yeah, we firstly had to come up with where are all these plants distributed throughout the landscape. So we made use of the ACDAT and the South African National Vegetation Database. These are huge vegetation plot databases that we have freely available from SANBI. So we got a whole bunch of vegetation plot data. I think in the end we ended up with about 10,000 that we used to feed into our model. And then we obviously looked through what plants we found in those plots, and for each of those we assigned them at the genus level. So our plot data was eventually aggregated at genus level. So we knew, for example, this genus covers 50% in this plot. And then for each of those genera, we assign them to either C3 or C4 or CAM. And I just want to stress, we're only focusing on grasses for C3 and C4, and for CAM, obviously, those are generally shrubs. So, yeah, okay, let's move on from there. 2 minutes, okay, I'm going to rush. No, let's go, Jasper. Then we want to obviously understand how does the climate control, like I said, these things respond differently to rainfall and to temperature. So we used the downscaled climate model that you've seen some examples of it from the various talks for current day and also for the LGM climate. And basically we used the climate data from that to construct bioclimatic variables. These are like bioclim, the guys who are involved with species distribution modeling. It's like the basic bioclim variables that you come up with. I've just highlighted the ones that we ended up finding to be the most important drivers there. I won't go into that though. We then applied paleo distribution modeling. It's basically species distribution modeling where we feed these different those three databases that I just showed you, the climate, and then the cover of the different photosynthetic types. Feed that into our models, and we used a random forest modeling framework for that. And at the end, I just want to stress our response variable that we get out from our models at the end is the relative cover of C3 and C4 grasses as a proportion of total grass cover, and the relative cover of CAM shrubs as a relative cover of total woody cover. Okay, so this is not based on species numbers or anything like that. This gives us a better proxy or a surrogate for the actual biomass of these photosynthetic types that we find in vegetation. So, what did we find? Firstly, just to show you, this is what we find for current climate. No real surprises. This lines up with what we expect, and this all lines up with vegetation surveys and things that we know in the literature. So I'm just going to move on quickly to this. This is a map that's been used quite extensively in South Africa and also for paleosciences, produced by Vogel et al. And it shows the relative cover of C4 grasses in different veld types of South Africa. And I just wanted to show, it correlates quite nicely, even though this is based on species numbers and not relative cover of the C4. We pretty much get very similar patterns when we look at that. And for CAM, again, nothing unexpected. We see nodes of very high CAM here in the southern Namib and the northern GCFR, Knashflakte, and then going into the winter, the valleys in between the Cape Fold Belt mountains. And just to stress, we have globally, these are some of these areas up here they're probably quite unique in that they have extremely high cover of you're making me so nervous, Jasper, of succulent caps, succulent plants that employ CAM. Okay, I want to just get through this last glacial stuff. So just to show you, we found again no major changes in C3 and C4 grasses. So C3 on the left, C4 on the right. Down here shows you the change from today going back to last glacial maximum with major changes in yellow, major positive changes in yellow. Gray areas indicate no change, and then the blue is where there were reductions. So in these uplands is where we find the major changes. So reductions in C4 grasses and increases in C3 grasses. But if you look at the overall distribution of those changes, it's slightly skewed towards sort of a net gain for C3 grasses. So overall, we find more C3 grasses in these during the last glacial maximum, but it's mostly concentrated in these north-south trending Cape Fault Belt mountains. And then for CAM, we find, again, if you look at the distribution of the changes, it's sort of distributed around 0. So overall, we're sort of finding the same level of CAM that we do today, but there were obviously some landscapes where we had some reductions. But again, CAM doesn't completely disappear, and this area up here was sort of a bit of a refuge for these high CAM nodes. Okay, so what does this mean? What do we care? Firstly, we just want to show off that these maps are probably much more useful compared to this map. Firstly, because it's not based on species numbers. We have a much more accurate surrogate for the actual importance of these C3 or C4 grasses in the vegetation. And secondly, it's our maps are produced at a much finer spatial scale. So these were produced at 1 in 500,000 scale, and the patterns are extrapolated to these very, very broad felt types, whereas ours are, they're much smaller based on raster grids. And then lastly, just to summarize or to conclude, is there weren't terrible, huge upheavals in distributions of our photosynthetic types, particularly the C3, C4 grasses on the south coast, this Paleogallus plain. So we had these mixed C3, C4 grasslands in that area, possibly like the stuff you see at the back there. And these grasslands or these grassy vegetation types, it would have been a Renosterveld and these alluvial plains. Because they had C4 grasses in there, it was quite likely that they would have been able to support these herds of large herbivores, which would have given these guys some protein to rely on. All right, I'm going to wrap it up there. Thank you very much.
I think you need to have a question though.
No questions.
Adam's got a question. He likes isotopes.
I know.
One question from Adrian.
Great, I have a question.
Hi, thanks. I'm just curious how you handled CO2 in the predictions of C4 and C3 grass distributions in the LGM. Just with the context being that you're now at 180 ppm CO2, which is like almost compensation point for C3, and how that would influence estimates of productivity, etc.
So this is why I didn't want to take a question. But anyway, so the major limitation of this model is that we haven't accounted for the lower CO2. It was about half the level of what we're seeing today. So particularly for C3, C4 grasses, that would have definitely made a change in what we would expect those patterns to look like. Under those conditions, the C4 grasses would have been able to outcompete some of those C3 grasses. So what we expect is to probably find, so Richard and them actually did in their paper in 2020, they did a temperature crossover analysis for the C3 and C4 grasses, and they found that along the Southern Cape and on the Paleogallus Plain, for most of the year, the temperature regime was enough to allow C4 grasses to actually outcompete C3 grasses. So we'd probably expect to find a higher signal of C4 grasses than what this model predicts at the moment. Yeah. Yeah. Sorry, I'm happy to chat later, but we'll move on from there. Thank you. I'm going to take over as chair.
Shorter than Adrian. Okay, so hello again. I'm going to move from my previous talk a little bit to the South Coast, and I'm a little bit bummed that I didn't talk more about to Adrian about what the outcomes of his study were, and you will find out why. So in this talk, I want to talk more again about the work I've been doing on the South Coast. So Curtis mentioned a publication by my PhD advisor, Miriam Barr-Matthews, that was published in 2010, which was basically the first speleothem record that we had from this area since the early 90s. And since then, I have kind of taken over the South African speleothem work from her, kind of in this group, working with Curtis and Richard, of course. And I actually have published 4 or 5 speleothem records from the South Coast now, but I'm going to focus on a kind of a new record. I wanted to show you something that is not published yet. So the motivation for this paleoclimate work on the South Coast in general is probably, we want to kind of know what the climate on the edge of the Paleogallus Plain was like as compared to other regions in South Africa. And in this case, I'm basically looking at the Little Karoo Basin. And the reason why we're interested in that, of course, is because we have pretty steep gradients in the present-day climate and topography between these areas. We can also say a little bit about vegetation history. Of course, we have this extraordinary diversity and very high endemism in this region, so we want to know what happened in the past there, or how we, what might have been going on. And then, of course, we have a 200,000-year inhabitation history by modern humans, or humans. And we want to know what kind of environment they lived in. What I'm going to talk about today, or in this talk, is a comparison between a site that we call Sandkraal Cave on the south coast to a site called Eflax Cave on the that is located on the southern foothills of the Swartberg Mountains. And here's a topographic map. The bathymetry is also kind of indicated, so the lighter colors basically show you where the Paleogallus Plain would have been located. So obviously, these environments would be quite different. So currently, Sandkraal Cave is literally 10 meters above sea level. But of course, as Haley has pointed out very broadly, is for 90% of the time, that sea level would have been much lower in the last 500,000 years. So there would have been a coastal plain outside of that cave that would have been varying over time. Whereas Eflax Cave is currently located at about 700 meters elevation. And it is separated from the coast by the Outeniqua Mountains, which are about 1,000 meters high. So there's first a mountain range, or the first mountain range, then there's the Little Karoo Inland Basin, so with some lower elevations, and then we get into the foothills of the Swartberg Mountains there. When we look at the climate, obviously the rainfall amounts at Eflax Cave will be a little bit lower than on the coast, but we still get about 500 millimeters. Sandkraal Cave is actually located quite close to where we get this real peak in seasonal rainfall on the central south coast. So we have about almost 900 millimeters of rainfall there. And of course, the seasonality of the rainfall is kind of a year-round aseasonal rainfall at both sites, maybe with a little peak in the transitional seasons. the vegetation at both of these sites is what we kind of consider part of the Cape Floristic region, but of course there will be differences because of the differences in climate. So at Sandkral Cave, I think above the site we have a kind of granite FYNBOS. It's very altered though, because there used to be a farm there, and on the slopes we have strandveld vegetation. And then at Eflux, we actually have quite a mosaic of vegetation types in the Little Karoo. And a lot of it depends on the aspect of the slope. So we will have more dry adapted vegetation types on the northern slopes and sometimes more FYNBOS or thickety kind of vegetations on the south exposed slopes. And Eflux Cave is actually located under a north exposed slope. So we would expect to see that reflected in our isotope records. And then I am showing the old map of Vogel here for the abundance of C3 and C4 plants. And it's just to give you an idea, and I think Adrian showed this much better, that on the south coast we're kind of looking at a 50-50 distribution of C3 and C4 grasses, whereas once we go inland to the Eflux site, C4 grasses are much more likely to occur there because we're much closer to these dryland grasslands that we find in the inland areas. Okay, so I talked about this a little bit this morning, but I just wanted to mention it here again. So we can date our speleothem records with uranium thorium up until 700,000 years ago. In this area, we really think that the oxygen reflects seasonality, with higher values usually referring to summer rainfall and lower values more likely winter rainfall. And we did a 4-year monitoring study in Mossel Bay that showed us that quite clearly. And then we have the carbon isotopes, and we oftentimes interpret those in terms of C3 versus C4 plant proportions. And I'm also, in this case, presenting a composite of multiple samples from the same site. So each of these sites has between 5 and 7 samples. And basically what I did to make these composites is I just combined all the data that I had from each individual site. So I showed you, for the Robertson record, I showed them individually as time series. So I would take a data set like that and just combine it into one table and run a smoother through that to get a single data set for each cave site. Okay, so here's the results for Sandkral Cave. Again, the age in the bottom goes here from about 10,000 years ago is when the record, the youngest part of the record, and the oldest part goes back to about 140,000. I do have to put in a little caveat here, because we had these changes in sea level. The cave probably was open at the transition from the last glacial stage 6, which is indicated at the top, into stage 5, sorry. So the first part of this record might not reflect environmental change, but it might be related to in-cave processes. But I think from about stage 5C onward, we have a good replication of this record through sampling multiple samples. And that usually indicates that we have an environmental signal rather than an in-cave signal. Okay. Then comparing this to our inland side, there's quite a steep offset between these locations. And what we think here for the oxygen isotopes, most likely this is just basically a signal of moisture rainout. So when moisture moves from a coastal area to an inland area and rain starts falling out of it, that usually removes the heavier isotopes first. So we get this shift towards lower values, which indicates more lighter isotopes in that rainfall. And what we see is basically the signal was more or less constant through time, because the offset seems to be more or less constant. And then for the carbon isotopes, the pattern is, of course, the opposite direction, which probably reflects the higher C4 grass abundance at the inland side. Okay, so we have mentioned this study by Engelbrecht et al. With the last glacial maximum climate reconstructions. And here are some maps showing basically the changes in rainfall, seasonal rainfall between the last glacial and the present day. So the map on the left will be a change in summer rain, and the map on the right will be winter rain. So what we see is basically for this study area on the south coast, we had an ever so slight increase in summer rain. But then we also see that there's quite a significant decrease in winter rain. And what they inferred for this is that it is due to a northward shift of the westerlies, which then causes downwind effects along the coastal mountain ranges, which leads to the more frequent occurrence of these adiabatic föhn wind conditions, which here is called berg wind. And what I wanted to look at is, for the time periods that I have covered with my records, do we see a similar thing for other cold periods in the past? So I have now at the top, again, I have the marine isotope stages, and I have kind of little asterisks next to the periods that are generally considered cold intervals. And I made Z-score normalizations out of the speleothem records. So they're now kind of plotting together around a zero value. And I think we can see, of course, there's always a lot of noise in these kinds of records, but I think we can see that, for example, during stage 5C and stage 5A, both the oxygen and the carbon isotopes tend to be lower or below zero. And then once we go into stage 5B and stage 4, they tend to be a little bit more above zero. So we basically can say that our interpretation of this, with having more winter rainfall during these warmer periods and more and less winter rainfall during the cooler periods, does apply. And the same for the vegetation, which of course contradicts what Adrian just said. I think that for the most part, there tends to be more C4 grass in this area during cool periods than during warm periods. Okay, and those are my conclusions. Thank you very much.
Thank you, Kerstin. That's blinding. We can do one 10-second question. Jasper Slingsby.
Now I get nervous.
No, this probably isn't a question for you necessarily. It was just, I mean, so in this inference around the C4 grass thing, I mean, is there nothing from the caves? I mean, most of the C4 grasses would be useful as fiber for things. I mean, things like phytoliths or that kind of thing.
So we do have a phytolith record from the archaeological sites at Pinnacle Point. And I think they corroborate this finding pretty well. The resolution there is quite a bit lower than what we have for the speleothem record. But from what I remember, you know, they show us very similar signal. I haven't looked at fire proxies in my speleothems, but there's most likely also, you know, options there to do that. Okay.
Thank you very much. Our next speaker, Richard Cowling.
So I got into this project for 3 reasons. The first one is the people involved in it were really nice people. There were no liver eaters. The second reason is I've always had an interest in the historical biogeography of the Cape flora. And these guys had the means and the funding to generate these incredible records that we wouldn't have been able to do without a huge input of foreign funding. And the third is there a third? Probably not.
All the point breaks.
Yeah, all the point breaks. Okay, so my topic is the vegetation of the Paleogallus Plain and what its significance is for the evolution of our coastal flora. And what I mean by the coastal flora are those species that are adapted to calcareous soils. So I'm excluding the species associated with the older leach sands that generally occur interdigitated with this more calcareous flora. And I want to recognize some of my friends, people who have played an important role in the development of this project. I also want to say that the papers that I refer to here have been published now. When we prepared the symposium in 2020, they weren't. So if you want to go and look at the details, they're in papers in that special issue of Quaternary Science Reviews. So, the two major floras that we're looking at, or vegetation types, are dune fynbos thicket mosaics, commonly known as struntfelt. We've seen it, it's all around us. And then also the limestone fynbos, the species associated with calcarenites, cemented sand dunes. And what's amazing is that these are incredibly rich floras. So, they're very rich floras, and the bottom line is that compared to other Mediterranean climates, they're extraordinary. So if you look at so-called struntfelt in north of Santa Barbara, the area-adjusted diversity there is 4 times lower than Langebaan. If you go to Western Australia and look at what's on their calcarenites there, it's a miserable bloody flora. So what's going on here? What's going on? The domain that we looked at for the study was the coastal plain, everything south of the mountains, and really from about Walker Bay to the eastern end of Algoa Bay. And that gives you the major vegetation types in the area. The yellow thing, bits, polygons along the coast. The bright yellows, Holocene sands. Okay, fairly recent calcareous sands. And the lighter yellow is calcarenites. Now, calcareous substrata only occupy about 4,500 square kilometers, or 4% of the Cape Floristic region. And it's equally shared between dunes and calcarenites. So it's a tiny little sliver. In some place, these dunes, areas where dunes can't accumulate, where you have steep topography, these dune areas are about, a couple of 100 meters wide. And you walk in them and they're fraught with species. And I live in one of these areas. And I've always been boggled by how rich they are. So this is work from Tony Verboom and his collaborators. So what we have here are some Cape lineages. What we have there is number of branches, which is giving you some idea of when diversification occurred. And what we have here are lineages associated with different substrata. So we're interested in those associated with calcareous substrata, which are orange. And we can see that those, along with the lowland sands, represent the latest diversification of the Cape flora. The latest diversification event has occurred on these substrata when they started developing in the Miocene, when we started getting sea level fluctuations and the accumulation of these dunes. So it's a relatively recent diversification. And thank you, Tony. That's an extraordinary contribution. So the Cape flora's youngest radiation. Now, on the This side of the panel, we just have some endemics associated with dunes. The other side of the panel, we have some endemics associated with calcarenite or limestone, as us ecologists call it. And phew, it's a beautiful flora. And there are lots of them. So in this calcareous flora, this is work that Adrian and I have done, published recently in Peer J. There are about 1,300 species. About 60% are endemic to the Cape, so there are things from other regions, other substrata that get in, but you know, that are non-Cape, but it's essentially a strong Cape flora, dominated by Cape lineages. 508 species, about 40% are calcareous endemic species, and this is equally shared between dunes and limestone. So 40% substratum endemism. And you know, you can go into patches of limestone FYNBOS where 90% of the biomass comprise calcicol species. I haven't seen that. I'm sure it exists somewhere, Fernando, in the Mediterranean basin. But phew, when I went to Australia, again, they're miserable calcarenites, a couple of endemic species. And you know, why? And there are many highly localized endemics, especially in the dunes. And what happened with sea level fluctuations, little populations of things have become stranded on these little remnants of what we'll see and what Haley showed to be a massive dune system. Okay, so we've seen this sort of stuff before. I don't have to elaborate on it, but at a full glacial, the Paleogallus plain was about the size of Ireland, or approximately the size of the extant CFR. So if we want to think of the extant CFR over, you know, 90% of the Pleistocene, it was somewhere between, who knows, I mean, as Haley said, things fluctuate, but at its maximum extent, it was double its area today. It's what Steve Hopper would call a yodfell. But we can see that because it's a young, frequently disturbed, relatively fertile landscape. Okay, so we did a vegetation model, and I'm really not going to go into much detail about this because it's all in the paper. But we, based on Haley's geology map, we developed a soil map using analogous soil data from the onshore region. And, you know, interesting things, as we've said, is a large area of this Paleogallus plain was dominated by really rich Cretaceous soils, the kind of things you get in the Gamtoos Valley or around Riversdale today. And the distal portion had vast areas of calcarenites, there were dunes, blah-de-blah-de-blah. We divided these things up into different soil categories based on soil depth and fertility. So soils more fertile than those on the contemporary Cape floristic region overall. We also looked at climate, and you know, on the left panel you can see, I think, God, it's hard to work this out. Anyway, let me give you the bottom line, okay? The bottom line is that the LGM daily maximum temperatures were 3 to 4 degrees higher in the midsummer and only 2 to 4 degrees lower in winter. And that's possibly because of the impacts of the Agulhas current being so close to the shoreline then, and which likely strengthened during glacials. Okay, so Paleogallus plain was also frost-free, and that's very important for vegetation modeling. Looking at the climate, again, let's just get to the bottom line here. There was more rain than present in the winter rainfall area, and a slight eastward expansion of the summer rain in the east. So the proportion of summer rain was much higher than it is at present in the east during the last glacial maximum, and lots more rain in the very western section of the Paleogallus plain, as you can see with the dark blue concentrated in that area. Okay, so more warm seasonal rainfall in the east. And we did take carbon dioxide into consideration. Don't try and comprehend that. Again, what we did is during glacials, when carbon dioxide levels are lower, atmospheric carbon dioxide levels are lower, water use efficiency declines. So, plants have to use more water per unit of carbon gain because there's just less carbon dioxide in the atmosphere. But at the same time, there's also lower vapor pressure deficits because temperatures are overall slightly lower. And we balanced these two things and did a whole lot of simulations, blah, blah, blah. And what we found was that the model accounted for low carbon dioxide and low water stress. And that enabled us to shift the thresholds to adjust for carbon dioxide in the vegetation model. So to summarize, the vegetation was based on soils, temperature, climate, carbon dioxide, and of course the big one, which is fire. So Tineke Cry, also in the paper, in the special issue, modeled fire incidents through fire danger days or whatever, using contemporary areas and then extrapolating it out onto the Paleogallus plain using the LGM model. And what we found is that here you have your index of fire intensity. It can be, I think that one is mean monthly FDI, and this is the number of FDI days greater than 12. And along here we have months starting in January, ending in December. And the modeled stuff is in black, and the model stuff is in black. The actual information is in either dotted lines or slightly less dotted lines. Anyway, I'm sure your brains are swimming. But let's just get to the bottom line again. The severity of fire during the LGM across the PAP was significantly higher than in the past. Okay, so our models could really do well in explaining extant incidents of fire. So we were quite comfortable extrapolating them. And you can see that fire danger indices during the summer months were extreme, probably as a result of increased wind regimes, lower humidity, and higher temperatures. So the Paleogallus plain burnt like hell. And there were humans out there, and as we'll see from Kirsten's work, who were really incentivized to burn it. So then we created a vegetation map. So at the top, we have the current Muchina and Rutherford vegetation map. The second panel is the vegetation, Muchina and Rutherford's vegetation predicted by our model. And we did all those AUC nonsense and what have you, and they were really good fits. We did it for each vegetation type separately. Obviously for FYNBOS and the types that are so strongly edaphically bound, we got good agreements. But there's some interesting things, which I'm really chuffed about, is that we predicted grassland in the eastern Renosterveld, east of the Breede River. And when you go and read the old accounts of the travelers, they all mention this Blaugrasveld, dominated by Thermida, and it's been trashed and converted to Renosterveld, but real Renosterveld west of the Breede. And a few other little idiosyncrasies, but basically our map did pretty well, or our model did pretty well in predicting the extant vegetation. So we were very happy to extrapolate it out onto the Paleogallus plain. And what Haley told you, there were vast areas of dune field, also large areas of grassland. We predicted, taking into account those thresholds with carbon dioxide and the incidence of C3 and C4, that they were mainly C4 grasslands, certainly in the eastern section. And down at the distal end, huge chunk of calcarenite, limestone fynbos. And all these vegetation types were dissected by these vast plains which were full of alluvium. And most likely, given what the extant situation is, supported a savanna, Vachelia Karoo, Buddleia, blah, 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 maybe some riverine gallery forests in places. Beautiful habitat for large mammals. But the thing that interests me is relative to today, there was a 23-fold decrease. Today, looking at our dunes, they are 23 times less extensive than they were at the LGM. Okay? And there was an 8-fold decrease in the extent of limestone fynbos since the LGM. So these vegetation types or floristic biomes were widely, hugely distributed across the Paleogallus plain. And what we see today are the remnants, particularly of the dunes, that have really been constrained to tiny little areas. The few areas where dunes can accumulate, Walker Bay, up here on the west coast, where I live around Cape St. Francis. But for the rest, all you have is tiny little slivers that are left. So, we can get back to explaining why this flora is so diverse and so rich in endemics. It's a legacy of historical phenomena. Now, this slide I'm going to miss. Okay, it's published. You can go and read it. The bottom line, okay, I'll give you the bottom line. Limestone flora, it's a species area curve. Log area on the bottom. How's my time? Oh, okay. Okay. Log area on the bottom, species numbers on the y-axis. Adrian has explained it. But limestone floras, lowland floras on sandstone are about 1.8 times richer in the east, in the west and the east, which, you know, there's a lot of data to corroborate that. But the sandstone floras were only 1.4 times richer than the dune floras, which, you know, these sandstone areas are ancient, yet these dune floras have diversified to the point where they're not that much poorer than sandstone floras. And we found no difference between the eastern and western dune floras. I think that's because there's an incursion in the east of subtropical taxa and lineages. And in the west, there's probably been more diversification and less extinction. So just summing up, the PAP contributed to the youngest radiation of the Cape flora. This dynamic is well understood. You know, we understand sea level changes and how much was exposed and all the rest of it, whereas in many other terrestrial systems, we don't have a dynamic that is so well tied down. And I think the time is ripe to focus on a montane versus coastal adaptation on the Cape of the Cape flora. So, for example, what are the differences in physiognomy, physiology, and what have you between Cape lineages that have radiated on the recent calcareous substrata compared to those in the Cape stronghold, its ancient stronghold in the mountains? I think we could learn a lot about phylogenetic ecophysiology if we went in that direction, and I urge my colleagues who do this kind of stuff, here's a great opportunity. Thank you.
Thanks very much, Richard, for that. Yeah, well, I'm super biased, but very interesting talk. I'm unfortunately going to...
We do have a discussion session.
Yeah, we have. So I'm going to suggest...
At the end. So if you've got some questions,
yeah,
please.
I'm going to suggest that we keep questions till the end for all our talks. Yeah, and then we can have a bit of a chat there. Who is next? Kale. It's you. Okay.
Okay. Hello, hello, hello. That's about the right distance. All right, I'm going to step back from the Cape Floristic region and look at essentially an intercontinental comparison that hopefully sheds light on what you see here and maybe extends the dynamics that are being inferred in this session in terms of long-term drivers of plant biodiversity gradients. So this is a study that is using something a bit unusual you probably haven't encountered before, which is getting fossil pollen records out of dirty speleothems, out of dirty stalagmites. This is a 1 centimeter slab from a stalagmite from one of the caves I'm going to talk about, Ecology cave, which is about 3 hours drive north of Perth. And these are well-ventilated caves growing in Pleistocene aeolianites. And because they're well-ventilated caves, as they grow, they accumulate all sorts of dusts, charcoal, pollen, fungal spores, and silts and clays and so on. And so they offer the potential for relatively precisely dated records from times in the past when rainfall was high enough to grow stalagmites in these regions, which is at various times over the last few 100,000 years. And this is showing a picture of this slab that's been marked up, getting ready to be cut with a bandsaw into relatively thin layers for dissolution and then pollen analysis. So this is the framework here. This is a question Richard and I have been talking about for about 10 years. He's been thinking about it for a lot longer than that, about, in particular here, not so much this global question of why these southern hemisphere subtropical regions have extraordinary plant biodiversity. We've heard a bit more about that from Adrian earlier in the week, but in particular, I want to focus today on this east-west difference that we see on both continents. So actually start with the right panel there, which is an extract from Cowley and Lombard, where they drew attention to this difference in area-adjusted species richness between the southeastern part of the Cape and the southwestern part of the Cape, with very distinct regression lines running through those, showing this striking pattern. And on the left, there's an extract where we did some compiled data in the Australian context. So you see the white circles are, this actually I should point out that on the left, this is just restricted to woody species. So I've excluded herbaceous species from this, which penalizes the Cape, but it doesn't really penalize Australia so much. So the white circles are Australian data that show from southeastern Australia, and the regression line through those, and the yellow diamonds are Southwest Australian occurrences, species richness in different size plots. And you see that then the purple ones are corresponding plots for the Cape Floristic region, and you see that although Richard likes to talk about how shit the Southwest Australian flora is relatively, if we exclude all your bloody geophytes, then they're really quite similar in diversity, right? So the woody components are really similar. Okay, you've got 12,000 geophytes. So you see there's something going on there, and we're interested in woody diversity because there's probably a different history, and a lot of it's more long-lived and so on and so forth. I can't really got time to go into that. And maybe we're biased in that way in Australia because maybe our flora is on average, a bit older. So we have a lot more, or whatever's going on there. We didn't really have time to talk about it. But so that's the starting point for this, was trying to explain this. Now, there's all sorts of neo-ecological ways that people have tried to explain these patterns, both at global scale, of course, energy theory and so on. Very few people have tried to explain it, the east-west gradients on both continents. Not many people have tried to explain that, but most of the explanations that do exist are based on kind of neo-ecological things or neo-climatological things. So, but since I'm a paleo-environmental person, of course, every problem, you know, I've got a hammer, every problem is a nail. So yeah, I see a nail and I've got my hammer. So to me, paleo-environmental data provides some insights that neo-environmental approaches simply can't look at because they don't have any access to the long-term evolution of various systems. And so that's the unique insight of paleo-environmental records. So this is a very well-known record. I did my best to try to make this more presentable for PowerPoint by taking this into Illustrator, but this is from Parkington's paper from Elands Bay, which is a rock shelter that was occupied at the last glacial maximum. And this various vegetation types divided, you know, into percentages of the whole. So that record actually has a charcoal, macro charcoal record identified often to species level, but also has a pollen record. This is the pollen record. And the takeaway message from this site seems to have been that it shows a wetter last glacial maximum than today. Basically, that's mostly motivated by interpreting that turquoise bottom, that dark turquoise section of each of those lines, which is scrub forest or afromontane forest taxa, which perhaps were growing in sort of scrub kind of form at the time, given the low CO2 of the last glacial maximum. But you see that the time step in which they're best represented is that 20.5 thousand year one at the LGM. So that's a very interesting premise that up the west coast, at least, in the winter rainfall climates of the Cape, last glacial maximum climates seem to have been wetter. It's difficult to conclude anything else from this record, wetter than today. But the limitations of this are, well, it's really very poorly dated. A lot of old archaeological records are just very poorly dated. It's not very well constrained. How long did that vegetation type persist there? Whatever, who knows? Also, the climate has never been quantified from this. It's just a bit of a qualitative hand-waving. And there are just few comparable records to compare that to. So I'm going to move now into the Australian context where we can address some of the same questions about east versus west using speleothem palynology. But to orient you, I'm going to show you two very summarized ordinary lacustrine conventional pollen diagrams that are in the Australian literature to fix you on what I call the Australian, it's a more widespread, but I call this the glacial aridity paradigm. If there's any place on Earth that doesn't have the glacial aridity paradigm strangling you by the throat, it would be South Africa. But everywhere else on the planet where there are trees, we are at the beck and call of the unquestionable glacial aridity paradigm. Now, mainly, there's two sources of inspiration for what I call the glacial aridity paradigm, and they've been in place for about a half a century. One of them is dust, and I won't go into that, but I can talk to you about it later if you want. But the main one really is pollen. So that's great, that's right in my laneway. And the gist of this is basically, and you can see this in both of these records, they're very poorly dated, but they basically show the black curves are trees, often eucalypts, but sometimes combined with a few other taxa in both of these records, and they have high tree cover through the Holocene, so through the current interglacial climate with high CO2, that's the CO2 curve on the red there, on the right, and those purple lines are approximating where the Pleistocene-Holocene transition probably is in those pollen records and precisely where it is in the CO2 record. That's from air bubbles in ice cores in Antarctica. And we basically have this repeating pattern all over the world, and certainly all over Australia, where we get vegetation structurally similar to today, right through the Holocene, and then in the pre-Holocene, and especially at the glacial maximum, which is a bit hard to pick out probably in both of those pollen records, you have basically treeless assemblages. So that's so the trouble here is that those treeless assemblages were looked at by geologists, paleo people, trained in uniformitarian methods, and they said, well, where can you find treeless assemblages today in this landscape? And inevitably, those were in the dry ends of the environmental gradients. And so they said, well, these climates were, and from other evidence, they said these climates must be cold and dry because if you get these treeless environments dominated by Asteraceae and grasses, they must be dry climates. But there's this totally ignored emerging understanding of the plant physiological effects of low CO2 at the last glacial maximum, and as Richard happily introduced for me, this massively hits the water use efficiency of C3 woody plants in particular, and the result of that is that it's extremely difficult to be a tree and to grow a forest in competition with herbs and shrubs under low CO2. So basically the whole world looks drier during the glacial maximum, even if moisture availability was no different. The whole world would look drier, and you would expect that all biomes would shift in a dry direction under the CO2 that existed at the LGM. So I'm going to look at two stalagmites. I'm going to present to you quickly some diagrams, one from Ecology Cave, which is, as I said, in southwestern Australia, north of Perth, and there's some other little lines there from some other little red dots actually indicate some other caves in that region. And another one from Naracoorte Caves, a well-known megafauna vertebrate site in South Australia, in the southeastern region. Both of these sites have Mediterranean-type climates. Western Australian one a bit more strongly, but both of them are quite strong Mediterranean-type climates with 500 to 600 millimeters of rain per year. And this is the climatic moisture index mapped over parts of Australia, which I'll introduce to you in a moment as a way of interrogating these records. So this is Ecology Cave. It's north of the forests that exist in the far southwest of Western Australia. It's dominated by shrublands. This is Pleistocene. This is the crappy species-poor Pleistocene aeolianites in Western Australia that Richard referred to. It's not that bad, really, but that's on the right there, one of the short Banksia group with probably some acacias and at the moment I've forgotten, and various Myrtaceae, I think, with a number of other herbs and things. I mean, he's actually quite right. So a little bit, 20 kilometers to the east of this site, when you get out of the band of the Pleistocene aeolianite, which runs right up the west coast, you get into the laterites of the Eneabba biodiversity hotspot, and this is off of that and is definitely not as species rich as that region. So this is the pond diagram. It's made up of several different stalagmites. So in the Holocene, we have just snapshots from two young Holocene stalagmites, one that's about 1,000 years old and one that's about 6,000 years old. And then there's a gap down to the main meat of this site, which is this other stalagmite, which grew from about 25,000 years ago up to about 22,500 years ago, and I've sampled this about 15 samples or something like that. So I'll walk you through this to interpret what's going on here. So basically, I originally approached this thinking, oh, we're going to see all this fantastic biome stability. So I was a little bit surprised when I first started working on these that there's actually quite a lot of representation of what I would think of as the classic glacial taxa that really, they don't make any, you can't see them at all up here in the Holocene. But this is like daisies, casuarina, which at least is endemic in the region, so you may not understand them so well, but they're weeds here. A big one, or two of them are weeds here. And salt bushes and grasses and so on. So this is actually sort of the classic stuff. I would mostly interpret that as a pretty low diversity biome, or part of a low diversity glacial biome. But we also see better representation. All those in the purple square are taxa that are better represented during the LGM than they are in the Holocene, are not part of classic Southern Australian low diversity glacial or even Holocene assemblages. It includes several taxa which are characteristic of and indicative of persistence of Kwong shrublands, such as cycads and various 3-porate proteaceae, and even other indicators that are not particularly indicative of shrublands but are indicative of moisture, such as fern spores, and a range of other taxa, Pimelia, and so on. And then when we move into the Holocene, we see actually quite a radical transition, quite similar to what we see in a lot of diagrams where we get reforestation. This site doesn't seem to have very many trees right around it today, but there's quite a lot of eucalyptus in that Holocene assemblage, and actually quite a lot of Banksia, which is perhaps best explained by realizing that there are Banksias that are small shrubs and there are Banksias that are 10-meter high trees, and it looks like they are as sensitive to the Pleistocene, Holocene climatic and CO2 differences as what we might more normally think of as a tree, like a eucalypt is. So there's quite a radical structural change here, similar to the kinds of structural change we see all over Australia and in many parts of the world as we move from the glacial maximum to the Holocene. So we'll pick this apart climatically in a minute. This is the other site in southeastern Australia, a much more low diversity environment, an open woodland of eucalyptus with, that's probably bracken on the ground there and grasses. And the pollen record here similarly shows some classical, boring, low diversity LGM ubiquitous taxa, grasses and daisies and salt bushes. I actually accidentally left grasses out of this pollen diagram, but there would be a lot of them at the LGM. But then again, a number of taxa that are better represented in the LGM, and another transition into much thicker forest cover or higher tree cover, including some Banksias, tree-sized Banksia, Banksia marginata there during the Holocene. So we're getting these records, although they're unconventional, based on unconventional sediment type, they're still producing very similar patterns of structural change as we go from the LGM to Holocene. Now, I want to just stop very briefly, that's too much to read, but to explain the approach I'm taking to how to reconstruct paleoclimate from these records. So abandoning the idea that you should use vegetation structure to reconstruct climate as being an artifact, largely, of CO2, instead we're going to focus on finding climatically sensitive plant taxa from which we can bioclimatically reconstruct the climate at the time. But it's also worth noting that we can't actually reconstruct last glacial maximum precipitation. We can't really do it because any time in a climate in the past that was substantially wetter or drier, warmer or colder than today, the evaporation environment, as Richard implied, evaporation is very different from today. So at the LGM, if temperatures were 4 or 5 degrees colder than today, evaporation was substantially reduced, and therefore we didn't need as much precipitation to produce the same amount of soil moisture as we do today. So to deal with that problem, which really is never dealt with, it seems, I've focused on an index called the Climatic Moisture Index, which was developed by Wilmot and Fadema. It has some advantages over more widely used Thornthwaite-derived indices, which can go up to infinity, in that it's symmetrical, so it's easier to interpret. It goes from +1 if there's no evaporation to -1 if there's no precipitation. And so I reconstruct that. I'm reconstructing that by mapping occurrence data onto, like this, onto a climatic moisture index surface for Australia. So this is just an example of what I'm going to be doing. I take for the various taxa, often genera, that are in the pollen records, we go and get occurrence data, quality controlled occurrence data, and then map those onto climate surfaces, like that climate surface in the background there, to produce a climate envelope for each taxon in the fossil record, and then look for the mutual climatic range, essentially, of the climatic envelopes of the assemblage. So this is just showing that immediately you'll see that for that taxon, that genus on the left, Amperia, which is small shrublets, about 5 or 6 species of small shrubs in the Euphorbiaceae, that genus does not extend as far north as Wheelowaggee Cave today. And you can guess right now that that's going to tell us that to get that species, or one of those 5 species, up to Wheelowaggee at the LGM is going to require higher soil moisture than today, because that's moving down the moisture gradient to get to Wheelowaggee. On the right we have a genus of cycads that don't so extremely predict a wetter glacial maximum, but you can see that the location of Wheelowaggee is probably at the dry margin of the distribution of that genus in southwestern Australia today, and so it's going to suggest that climates at least were no drier than today. And that is pretty much what we find. So this is box and whisker plots for each of the taxa I chose to work with in the Wheelowaggee pollen record, and then their mutual climatic range defined with 2 different levels of certainty or confidence, whether you want a broad one, which is in yellow, or that sort of salmon-colored one that's actually no, the salmon-colored one is less conservative and the yellow one is more conservative. So what all I'm doing here is looking for the mutual climatic range is defined as the wettest minimum and the driest maximum of the occurrence data sets for each of these taxa, but defined sometimes, so rather than define them as the absolute minimum and absolute maximum, which can have some artifacts in them, I'm looking at whether there's like the 0.5 percentile of each of the data sets or the 2.5 percentile of data sets. The modern values are the red dashed line. So on the right, because we all think in terms of precipitation intuitively, is a precipitation space reconstruction. But as I said, that's actually really misleading. It really wasn't necessarily that wet because it was a much colder climate at the LGM. And on the left is basically all that data transformed into climatic moisture space. So basically the take-home message of this is that because of several of these taxa that need more moisture than Wheelowaggee can supply, than the climate at Wheelowaggee can supply today, this clearly indicates that the climate was somewhat wetter than today. If we move to Naracoorte, and by the way, all those little black dots are jittered the jittered occurrence data, jittered in a left-right direction, which has no statistical meaning. It's just there to show how much data is there in the distribution of it. And Eastern Australia has 10 times as much plant occurrence data as Southwestern Australia, so these plots look much blacker than the Wheeler-Wodgee ones. Here, what we see is that there's again a sense that the climate was at least not much drier than today, and probably a bit wetter. So there's a difference between these two reconstructions in the southeast and the southwest. The southwest one clearly shows a wetter LGM climate, and the southeast one shows a contradiction to the glacial aridity paradigm, but not necessarily a clear indication that was necessarily wetter. It could be a little bit drier. It could be unchanged. So these impressions, I'll note, are consistent with the most recent PMIP4 ensemble work, which is summarized in Kageyama's paper just last year. So these are ensembles of all the modeling groups, climate modeling groups that participated in the LGM experiment as part of the PMIP program. And it's consistent in the sense that, both in terms of like this use of a moisture index rather than looking at raw precipitation reconstructions. So if you look at the top panel, that's LGM minus pre-industrial precipitation worldwide for precipitation. So that shows slightly pinkish colors over most of Australia. Actually, South Africa, I'm sure you're all looking at South Africa, it's actually slightly wet. But Australia is slightly dry. But then if we move to, well, the only index which in this paper is provided as a surrogate for moisture availability is just simply precipitation minus evaporation, which is a bit different from my moisture index, but it will do. We see that Australia shifts from essentially being pink to being pale blue. So the reduction in temperature means that even though there was lower precipitation, the environment was actually moister. There was moisture hung around the landscape longer and was more effective. And of course, South Africa does the same thing. So it's consistent with this state-of-the-art modeling effort that there's a change of sign as we go from LGM precipitation to LGM moisture availability. We can have slightly reduced precipitation and still have slightly moister soils and moister climates, and that has huge ecological implications. Rarely is that quoted, whether people are working with proxies or with models that consistently just show precipitation. They probably ought to show indicators of moisture availability, since that's really what is actually perceived in the environment. So conclusions. These reconstructions indicate higher LGM moisture pretty unambiguously in the southwest of Australia. The southeast, it's still a very newsworthy result because it really contradicts the idea that the climate had half the rainfall of today or whatever, that it was a desert and so on. That's really not true, but so both regions contradict glacial aridity paradigm, but there still seems to be a gradient between the southeast and the southwest in terms of moisture availability. So that's broadly consistent with previous interpretations in the Cape of this gradient between the southeast and the southwest, but glacial aridity in subtropical southern latitudes is actually probably not really a thing. It's just a misapprehension as far as I'm concerned, and if you want to try to convince me otherwise, I'm happy to talk about it some more. I think it's an overstated paradigm. But basically, this still leaves open the question, because in a way there's a paradox here, which is that although there's a lot of, it could be drier, it could be wetter, the southeastern reconstruction from Naracoorte suggests perhaps a lot more possibility of no change at all relative to today. Whereas the southwest, the Wheeler-Wodgee reconstruction, clearly says, no, actually the climates were wetter than today. And is that stability? How does that, to feed back to my original question, how does that explain the accumulation, clearly the accumulation of so much more species richness in the southwest of both of these continents rather compared to their southeast? It's not, so it's feeding into something complex about permitting biome stability, but it's not necessarily climate stability that's doing it. It's actually in a way, it's different. It's actually wetter. So we might want to talk about that in our discussion section, but I'll leave it there, and I'm happy to take, well, no, I'm not taking questions now, but thank you for your attention.
Thanks, Cal. Okay, I'm going to hand over to Martina quickly.
Hi everyone, sorry to interrupt. Just a quick announcement before we go on to the next speaker. So this session is a little bit late, but so we're going to move the thematic session that was going to be in Hall 1 to start in Hall 2 in about 10 minutes time. So if you're a speaker, Shirk, Kindwin, Stewart, and I think Robert, There might be another person that I'm forgetting now. Please just move over to hall 2 for that. And we'll also play the e-poster session before you guys start. But that way we're still catching up to start with the online keynotes at 12:20. Okay, and there's then enough time for facilitated discussion. I realize 12 minutes is a short time when we're looking at your time frames.
Thanks, Jan.
Okay.
You're up.
Thanks. So the hand from the past meets the hand of science, and this is a talk I want to do about how we got to creating resource landscapes and then getting to the paleo landscapes. So the field we're in human behavioral ecology, we also employ ethnography, experimental archaeology, etc., can really complement the archaeology, especially if you have extinct landscapes and direct evidence is lacking. So we have the, as you've seen so many times already in the symposium, we have the extant landscape today, and then obviously you have the extinct landscape. We first use natural resource economies in calories to determine values of resource landscapes, which you just call resource scapes. And that then we reconstruct our extinct resource landscape, which we call paleoscapes. And as you've seen, we've already started with the Paleogallus plain, and as a conclusion so far, we can see possible increased productivity and resilience on the Paleogallus plain. So how do we go about doing this? In the field of human behavioral ecology, we do foraging experiments to determine kilocalorific averages for various resources in various vegetation types, marine habitat types for these different habitats. But it requires a few things in that habitat. We need people, indigenous people, with a knowledge that they've kept with them from the past, their heritage. Fortunately, in our country, we still have a lot of these people with a beautiful heritage, cultural heritage, that they've brought with them from the past. And secondly, we need pristine vegetation, we need pristine marine habitat types, depending on which resources are studied, to employ these foraging experiments. So let's start with the vegetation. I'm just going to run you through the various resources which we get off economies, natural resource economies from, starting with plant food, which I'm going to leave for Susan Botha, which is the next talk. I'm just going to quickly run over it. So we create a study area, obviously, employing Messina and Rutherford, and we have various communities that we take to the various vegetation types in their pristine forms, and then through our methods in our field, we have these foraging experiments which then capture the calorific averages phenologically over the year from January to December. Fortunately, when we started off this project, we had no idea of the knowledge base that existed with these people in the Southern Cape. And I started with ethnobotany, and it was amazing what these people still knew and effectively still ate out of their landscape. Just an example of fruiting species. And then obviously we saw some standouts in their knowledge base. For instance, with the Scyphia species, everyone there still eats it. As you've heard about geophytes, unfortunately some of the knowledge on geophytes has been lost due to, we think, the complexity of processing, the potential toxicity of some species, and as the knowledge started being lost, people became cautious and telling their offspring, their children, to stay away from certain things, and we nonetheless have a massive knowledge from them and also from the archaeological record from what's left behind, and we could design some foraging experiments around that too. This was quite a breakthrough. I picked this up in my ethnobotanical studies, that they eat the meristem of Bryonium serratum in the perennial streams, and Susanne's going to give you more about plant food foraging return rates. Then also, Susanne's busy with foraging experiments with fuel wood in the same vegetation types, employing the same methods. That's at the moment still ongoing, but very impressive. We didn't think the CFR had a profitable fuel wood load, but it's actually very impressive. And I'm not going to steal too much thunder, but also extremely resilient. So more on that at another occasion from Susanne. And then this is the stuff I'm more focused on in the Southern Cape, is the intertidal zone, marine resources, and I did the exact same stuff, methods. This is an example from just one forager. It's not the total harvest, but it's just an example of the various species that can come from the people when they forage. This would be in one marine habitat type called Table Mountain Group sandstones. We also then have aeolianite reefs. And just to give you one figure of my results, the y-axis is the kilocalorific return rates averages per hour, and the x-axis is the tidal level in meter. And that was the major predictor, or the most affecting variable on returns, on how much you can get per hour in calories, affected by the lunar cycle. I'm not going to get into that now, but most of you will know that in a day you will have a low tide and a high tide. When the moon is full or is no moon, you will have the highest tidal variation. It would be the most profitable time of day to forage at low tide, because then the most profitable tidal zone right in the front is exposed, offering the biggest prey and the most abundant array of prey. However, I looked at how does this compare to other places, and I'm not going to read them out, but my results was just on average just below 1,500 kilocalories per hour, and that's the highest ever recorded globally for, ethnographically from any community on planet Earth. So yes, the intertidal zone is extremely profitable, productive, and I'm not going to get into my resilience studies. It's also in the special edition of the 2020 QSR paper, my resilience paper, but the intertidal extremely resilient due to the mobility of certain species moving in from the subtidal once the intertidal is depleted. I call that the pantry hypothesis. 95% of those species that are mobile inhabit the subtidal. Then we also looked at fauna, various ways. I have a project with a student of mine, Haley mentioned it, we both his supervisors, Charles Helm, more than 300 sites, expanding our record from the body fossil record with the tracks of animals, also humans, potentially the patterns humans made in sand that has become aeolianite from the Pleistocene. We have the body fossil record, and there was experimental work done by Jake Harris, also at Arizona State University with Kim Hill and Curtis Marion. And we got encounter rates for fauna, bow hunting for vertebrate game, and we managed to also then layer our resource scape or paleoscape then with fauna and the return rates thereof. That then culminates on an agent-based model on the extant habitat, which is a virtual simulation of reality, and we have little agents or virtual hunter-gatherers roaming that landscape, foraging from all these resources which have been populated as layers on this model, terrestrially and obviously also in the intertidal zone, and we can then, through these models, as Curtis said, look at behavior of humans. But furthermore, I think, we can then extrapolate from the extant to the extinct, because we have the information, and what the PAP tells us is that we have a more profitable system, especially with the fauna. The intertidal zone showed that aeolianite reefs were the most profitable, and the PAP intertidal is dominated by aeolianite reefs. The faunal systems, as you've already seen in some of these talks, was amazingly rich with migratory faunal systems, some extinct megafauna today, etc. I'm not going to talk about the plant food. Susan's going to present on that now, but it's also quite impressive. Fuelwood, obviously, with expanded groves of Vachelia carew. And quickly, obviously, no problem with water. We see examples of calcrete lithics today in shell middens, digging stones, which ease the access to geophytes made of calcrete. And they obviously would have used bone and wood, as we see bone preserves in archaeological sites as tools. And overall, the Paleogallus plain was most probably a more productive and dependable landscape than we have today on the extant CFR. So we've built the toolkit to create resource-scapes and then extrapolate to paleoscapes, and therefore we can employ this toolkit to other habitats. For instance, we're busy with the Little Karoo, connecting agent-based models, these agent-based models to the Little Karoo, and then testing certain behaviorals, possibly trade, etc., between inland people, coastal people, and the list goes on. These models are actually very handy, and we've learned now to use these models for various questions with specific thresholds, etc. And the next one I'm also very interested in creating a resource scape for is the Paleo-Benguela plain. Thank you.
Thank you so much, Jan. Yeah, as I said, we're just gonna keep questions until the end. So we'll hand over to Susan Botha.
Hi, everyone. Yeah, so I explained to my sister what I do for research, and she said, she's in the film industry, she said basically there's only one place where people could have survived is under the water now, and you're trying to figure out what component the plants played in their survival. And I said, yeah, that's exactly right, except it wasn't a zombie apocalypse. And she was like, but you've got to spin it for a big movie, right? So there are many pretty flowers in the Cape flora, and my task was to see how you could eat them to survive. Sorry. So some of these slides might be a bit sensitive. So we focused on the south coast, because as Richard showed, all the vegetation types just got squashed onto the south coast, and I thought if I could figure out, spatially in the south coast, what the distribution of food is, we can then extract it to the Paleo-Gallus plain, which I'm going to call the PAP. And the idea is that, I can't remember. The idea is that we can then plug this into an agent-based model, right? So the foragers can then go into the PAP and we can see how they would have survived there, right, with this food data. And our research methodology is quite novel, that we use participants that grew up in these local communities. And that's quite important because I could have put out little plots and measured biomass, but humans make choices in the landscape, right? And there's selection time, there's search time, all of that. So it's more realistic to use people to do these experiments. And I had an amazing mentor in Jan. So I, over the period of 2 years, got into my little Mahindra, drove down to Stilbaai every month, met with these ladies from 9 different communities, and we really sampled 7 different vegetation types all across the south coast, 450 bouts in total, which makes it one of the largest published data sets with this type of work. And it was really quite fun. And I'm going to drive straight into the results. So we overall sampled 90 different edible plants, which is an order of a magnitude higher than any other thing that I could come across, but that's because the spatial scale was so large. But really, the mean calorific return is 140 per hour, which is really low, but really means nothing because these resources are so patchily distributed. So if you look here at the top left, you could really arrive in a lot of places on the south coast and find almost nothing. And then you sometimes hit these sweet spots where your calorific return is between 1,000 and 2,000 calories per hour. Now, supposedly a hunter-gatherer needed 2,000 calories a day to survive. And I think we're quite lucky when you walk out here in tea time with all those pastries, you probably hit that target much quicker. So there's not much seasonal variation, but there's another layer to the spatial variation, and that's in recently burnt fynbos. So if you come into a recently burnt fynbos area, you're going to get significantly higher. This is post-fire vegetation age, and that's calories per hour. So in sand and limestone fynbos, you're getting significantly higher returns if you're foraging on those landscapes. And what we then did, all the bouts that I did for all the vegetation types, I was curious to see if you were a forager, where would you go, to survive? It felt like that when I was walking in the field, like I have to eat today, So we randomly put those bouts together, ran 1,000 simulations, and the most profitable vegetation types are all on the left here, was sand fynbos and dune fynbos thicket mosaic, which is that strandveld that we saw on the field trip. Those two came out top. This is the hours it would take you to reach 2,000 calories. And it's really high. So for sand fynbos and strandveld, it's about more than 10 hours. So you would have to forage your little bat off, to make it every day. And when you go down to rhinoceros, it gets, like, I would say, unsustainable. That's between 25 and 50 hours. But sand fynbos, the reason this little vegetation type is so prolific is one species, Watsonia, and I think Anton mentioned they mass flower after fire, and Deacon planted this idea that humans could have maybe, used fire. So the problem though is, like, you come to this little clump of Watsonia and you're digging out corms, and you've got a little piece of bread in your hand, you can eat it straight away, but you can't, it's full of tannins. So a colleague of mine, she did all these experiments to see how you could leach the tannins out by soaking them in sea tidal pools, throwing them in the fire, and then just harvesting straight from a felt that had recently burned. And what you see here is tannin content on the left, right? And those are all the methods she tried. But if you don't do anything, the tannin content is really high. If you go to a place where there's been a veld fire, the tannin content is 0. So what I'm suggesting is that in Australia, there's the fire stick farming, where Aborigines would use fire to create a landscape where they could harvest food from. So, and I'm suggesting that there would have been an incentive for hunter-gatherers to do this in the fynbos. Cool, eh? So here comes the sensitive part. Obviously, you can harvest these foods, but they also have to be resilient. So can you go back to the same place once you've taken out all the geophytes? And I'd be interested to know what you think. Do you think you could? Well, my idea was that it's going to take a very long time, because it's like a little potato. Once you've taken it out, it's going to be gone, right? So we set up 19 permanent plots with the ladies, and I foraged them once a year, and I took out everything that was in that plot, and I was focusing on geophytes. Okay, and what I found was slightly higher returns in the second year. And that was really surprising. Only after 3 years of harvesting do I make a significant dent. And the reason for that is that only a proportion of the species population emerged in 1 year. So if you look here, those are, let's just look at this graph here, that's edible weight, and those are the species. So the different colored parts of the graph is, and yet the first year, I would take out everything with the ladies. We would take out everything, there's nothing left. Come back to the next year, and a part of the population has emerged. We would take all of that out, come back the third year, and there's a little part of the population that's still emerging, and we can take that out. So this is really amazing, and there's no documentation of this for geophyte ecology in South Africa. And it's probably a bed hedging strategy. But just one drastic example, in one of the plots I encountered Ferraria crispa, and we harvested all the above ground Ferraria crispa. And I really thought that I was going to come back the next year and find nothing. And the whole plot was covered in Cyphia digitata, which we then also harvested. So what does this mean? So we used this data, put it into the agent-based model, And then the little agents are designed to forage based on optimal foraging theory. And now they've got this data set. You know, they've got mammals, they've got shellfish, they've got food. This is on the south coast. And what they do, what the model shows that 80% of the diet was plants. But it was really tough going. So if you look here, that's daily calorific return on the left, okay, and population density on the x-axis. If you're harvesting for 4 hours a day, you're not meeting your 2,000 calorie threshold. You're not going to make it. 6 hours, 8 hours, only by 10 hours do you cap into that 2,000 calories. Only after 12 hours of foraging daily do you make it. So the South Coast was probably really hard going and survival, and that's what Curtis was suggesting with this PAP. You have this different landscape where this Garden of Eden, where we could have survived during glacial periods. So what I'm predicting is that once the PAP was exposed, I just want to show you the agent-based model was just covering that little area around Pinnacle Point. But you can see in that little square, there's a lot of different vegetation types, right, squashed in there. If you put that same size on the PAP, you might just find yourself in scrappy limestone FYNBOS. So this has got implications for mobility patterns. I mean, you've got to find food, and many of these vegetation types have different suites of species. If you, just because of the scale of the PAP, you might walk really far to find something new, right? On the PAP, you don't find any sand FYNBOS. You wouldn't have had that Watsonia resource, but you do have Strandveld, that dune FYNBOS thicket mosaic, and it's really abundant. And that returns the same calorific yield as sand FYNBOS. So if I was a forager on the PAP, I would have focused myself on the coast, because then you're also reaching into the intertidal, but around riparian zones where there's this huge abundance of Bryonia, Typha capensis, and things like that. And the only thing that is a concern is that during lower CO2 levels, during the glacials, we have a paper published with Karen Esler and one of her students. The bulbs make half amount, you know, they reduce their size by about half. So that's got huge implications for foragers. You know, you might have to work double as hard to get the same amount of food. And yeah, that's it. Thank you.
Thank you very much, Susan. Our next talk is going to be an online talk by Chris Brook.
Hello, everyone. Thank you for the opportunity for letting me present at Medecos this year. Unfortunately, I'm not here, but I hear that it's been an excellent conference throughout the week. So congratulations to the organizers, and thank you for this opportunity. So I am going to be presenting today on 2 of the chapters from my PhD, looking at using functional groups to predict the biomass and the probability of occurrence of large mammals across the Paleogallus Plain during the last glacial maximum. So to put everything into perspective, this is just what Pinnacle Point looks like today off the coastline of Mossel Bay. And you can see that it's this very sharp, rocky, jagged, fairly mountainous coastline. It's characterized by very unpalatable vegetation, FYNBOS vegetation, and we know from current experience that there's a very low density of large herbivores that you see in FYNBOS today. But that picture back to the last glacial maximum, to the last full glacial, around 21,000 years ago, and we get a completely different picture. Now we see an environment that's characterized by large open grassy plains. It's relatively flat, it's rich in nutrients, and it's this completely contrasting habitat, and it provides a very suitable habitat for large herbivores, and particularly large grazers, which coincidentally, we don't see in the Cape today. So just to, again, put this into perspective a little bit, this is an image from Maggie Newman, and it creates this kind of, this artistic representation of what was the Paleogallus Plain. You can see very high abundance of grass, large grazing herbivores. There's Sincerus antiquus, bluebuck, zebra in the background, and readily available water in this habitat. And this provides this idealistic environment, or this almost idealistic environment for large herbivores, which we don't see today. So what we aim to do is we aim to model the large herbivore biomass and drivers on the Paleogaze plain. We did this by firstly looking at species characteristics, so what are the behavioral and physiological traits of species, and what are the environmental traits, so what in the environment drives the distribution of species, and that's things like water availability and vegetation type. And then together species traits and environmental traits then feed into this, habitat suitability. How suitable is habitat? Does it create a landscape of fear for herbivores? And how does the distribution of resources influence the distribution of herbivores? And then finally that feeds into our model distribution and our model probability of occurrence. So we did this for 5 large functional groups. These functional groups were developed by Hemson in 2015. And it's 5 functional groups based on a body mass, diet, gut type, herd size, and water dependence. It's 5 of the main characteristics that then influence how these species are and what their functional type is. So where they're likely to be in the landscape and similar species they're associated with. And the 5 functional groups you have then is small non-social browsers. It's your Gracebok, Steenbok, Duiker, species like that. Medium-sized social mixed feeders. Your Springbok, Impala, species like that. Large browsers. Kudu, characteristic species, okapi, giraffe. Water-dependent grazers, so your wildebeest, hartebeest, species like that. And then finally your non-ruminants, so elephant, elephant, rhinos, zebra, species like that. And so then we modeled for each of these 5 functional groups rather than looking at individual species, because in a situation like this, when you're dealing with modern analogs for species, it becomes very difficult if you try and look at a species basis. So for simplicity, we just looked at the 5 functional groups. So firstly, what we had to do is we had to calculate, what is the predicted biomass of each of these species? And we did this for each of the biomes on the Paleogaze plain. And to do this, we used modern analogs from 39 protected areas in South Africa. And for each of these protected areas, we looked at, what is the reserve size? Reserve size strongly influences species composition and abundance. What is the rainfall? Rainfall is one of the strongest drivers of herbivore biomass, or it is the strongest driver of herbivore biomass. What biome did the reserve fall in? What is the percentage of winter rainfall? Obviously, there's a shift from winter rainfall in the Cape to more summer rainfall in the east and in the northeast of the country. And then we did this for each of the 5 functional groups across these 39 protected areas. Sorry. Then, secondly, we then looked at, what is the probability of occurrence? So we looked at probability of occurrence in relation to distance to water, and we looked at it for the 5 functional groups. We used data from Kruger National Park, being an environment also where water is readily available, similar to what we would expect to see on the Paleogaze plain. And there was very little differentiation between the 5 functional groups in terms of whether they would occur closer or further away to water, irrelevant of whether they were water dependent or not. And that's simply because even if you're a water independent species, if water is readily available, you're more likely to go and drink because it is there. Then we looked at, what is human risk? We looked at distance to humans, and we used a 14-kilometer buffer around human camps. And we did this again for each of the 5 functional groups, and we based, what is the risk of each 5 functional groups from the FETA 2020 paper, where we used a proportion of each functional group that made up the archaeological record. So in this instance, water-dependent grazers made up the vast majority of the functional group, so they were at most risk of being caught by humans because they made up because they were more prevalent in the archaeological record. And we did this according to 2 human densities. So we looked at high human densities and we looked at low human densities, and both of these were modeled sort of at the high and low end of what we expected, or the densities we expected to see of humans in historic landscapes. And then in these models, in these general linear models, we also included what is the vegetation density risk. Thicker vegetation is more associated with risk for smaller species. How does the landscape of fear influence species? Areas closer to water, areas in the steep riverine, in rivers where terrain is very steep, vegetation is very thick. These are considered more dangerous areas because it's easier for predators to conceal themselves and it's harder for prey to escape. We looked at vegetation type, herbivore biomass, which I explained in the previous step, and then for the 5 functional groups. So if we move on to our results, for small non-social browsers, the figure on the top left is how the predicted biomass increase or decreases rainfall. And you can see there's very little increase or decrease there, except in thicket. There is a slight increase. But if you look at the predicted biomass across the Paleogallus plain, it's extremely low. And it's extremely low because these species, these small non-social browsers, are more driven by behavioral mechanisms. They're density regulated rather than driven by the availability of food or the availability of resources in the landscape. So they're more restricted by their territory sizes and behaviors, which predicts a more even spread of species across the landscape, which is why we don't see any large contrast in predicted biomass there. On to looking at the probability of occurrence, then we see, okay, well, this is how habitat and things influence them. So probability of occurrence then jumps up in thicket and in some of the more dense fynbos environments. And this is, again, it's where there is suitable habitat for them. They are density regulated, but it is more idealistic habitat and habitat where risk is relatively lower than in other areas. And they were affected slightly more in high human density models, but it was a very negligible effect. The effect of humans was much more pronounced in high density models in areas where thicket was. On to medium-sized social mixed feeders. Medium-sized social mixed, they decreased in all environments except in fynbos, where there was quite a sharp increase. Medium-sized social mixed feeders were one of the water-independent functional groups. And you can see that on the far southwest of the Paleogallus plain is probably one of the only areas where water likely would have been very scarce. There's no big rivers, there's no big wetlands in that area, but it didn't affect the distribution of species. And a lot of those species, if you look at springbok, are species that are found at high densities or found at relatively high densities in fynbos and succulent Karoo and Renosterwald environments today. If we have a look at their probability of occurrence, their probability of occurrence then shifts quite strongly to more grassy environments. These would have been more open environments. There would have been environments where the landscape of fear, the risk of encountering predators would have been much lower. Grassy environments also would have had a lot more available forage, or a lot more available palatable forage. But in saying this, they still maintain relatively high densities in the fynbos environment. And in terms of humans, this was the functional group that was most strongly negatively affected in both the high and low human density models by the presence of humans. So the landscape of fear that humans would have created will have strongly negatively affected these species on the Paleogallus plain. In terms of large browsers similar to small non-social browsers, they're not species that you find at high densities. Although they did increase again in all biomes except in grasslands, which is what we're seeing in many of the other functional groups. But they maintained relatively low biomass across the Paleogallus plain. And in terms of probability of occurrence, suddenly you see this jump to more woody environments being preferred. It's preferable habitat. There's not as much risk to them because they're generally larger species. So you see in thicket a much higher biomass. In floodplain woodlands, you see a relatively high biomass. But in fynbos and grasslands and things, that biomass of large browsers is very low. And that, again, is what we see today. You don't see large browsers at very high densities in any of in the environments today in the Cape. In terms of water-dependent grazers, now water-dependent grazers, again, decreased with increasing rainfall. And I must make note here, it's important for this functional group that with an increase in rainfall in grasslands, there's a critical threshold around 800 millimeters. So palatability and herbivore biomass is likely to increase up until 800 millimeters and then decreased. But most of our sites were situated in grasslands above 800 millimeters wide, which is why we're seeing this negative trend in predicted biomass. And look at probability of occurrence. Again, you can see these grassy environments, so grasslands and floodplain woodlands have a much higher probability of occurrence. And interestingly here, this functional group was affected relatively little by humans, even though it made up the highest proportion of the archaeological record. So it's suggesting that, okay, well, these species are actually driven by other environmental factors. So driven by water, they're water dependent, driven by forage, they need to maintain a high quality grassy forage rather than the risk of humans in the landscape. Finally, if we look at non-ruminants, non-ruminants were highest probability of occurrence in, or the highest predicted biomass, sorry, in thicket, and this is likely driven predominantly by the presence of elephants in these thicket environments, large browsing species. But they show the same trend as many of the other functional groups where the biomass is increasing across biomes except in grassland. And then if we have a look at their probability of occurrence stays highest in these thicket environments, but it also increases in your more woody savanna type environments, so your floodplain woodland, and in your grassland. Grasslands then is predominantly driven by your large non-ruminant grazing species, so zebra and white rhino, things like that. And again, this carryover of species in or large non-ruminant browsers in the floodplain woodlands and thicket is where you're seeing their highest biomass there. So in conclusion, we've highlighted the importance, and it's well known and it's well published, these links between rainfall and herbivore biomass, and this can provide us with a very good estimate or a very good measure to retrodict historic herbivore populations. We've shown that early humans had the strongest influence on medium-sized social mixed feeders, and this is despite water-dependent grazers being the most frequent in the archaeological records, and it's likely that these are as a result of behaviors that you see these changes in probability of occurrence rather than a direct output from the archaeological record. So medium-sized social mixed feeders are being more affected by humans, whereas large water-dependent grazers, although they make up the highest proportion of the archaeological record, are more important, are driven by more environmental variables. And in terms of understanding historic trends, it's important we need to understand the current large herbivore dynamics, and we need to it's also important as it gives us this insight into, okay, well, how did early humans use large herbivores as a food resource? And then finally, the data from the study, the data from this project is then being fed into an agent-based model that's investigating early humans during the last glacial maximum and what they used, how they used the landscape, how they used resources, etc.
Thank you, Chris. Chris is not online, but yeah, thanks nonetheless for preparing the talk. Our final talk for this session, Curtis Marion is going to give us a quick synthesis of everything we've been speaking about, and just explain the importance of the Paleogallus plain for human origins, modern human origins. Thanks,
Okay.
Curtis.
Thank you. You've heard me say before that we've just completed a World Heritage nomination for the early modern human sites that we've been talking about a little bit. So you might want to ask the question, why are they so important that they would be World Heritage sites? And some of the important ones are indicated here. Pinnacle Point, which is a site that I've been leading excavations, is one of those sites. Deep Cliff and Sabudu are two others. So why? Why are they so important? We have the earliest evidence for coastal resource use at these sites, the earliest evidence for worked pigments at these sites at 162,000 years ago, earliest evidence for pyrotechnology, people using fire to manipulate materials, 162,000 years ago, earliest seashell collecting at 110,000 years ago. And when I say earliest, that's earliest worldwide. Okay, so we're checking a lot of boxes here. Earliest ochre paint, 110,000 years ago, even in a little shell at Blombos Cave. Earliest microlithic technology at 73,000 years ago. And microlithic technology is a breakthrough adaptation for projectile weapon technology, and it made the cover of Nature. Earliest engraved ochre, 70 to 60,000 years ago. Earliest decorated ostrich eggshell flasks, 65,000 years ago. So then the list goes on and on and on. I could actually move forward in this. And here's the age representation of those sites that I just mentioned. Time is on this axis, and these are the major sites. And the thing that I want to point out that's really important is when those sites were occupied, that plain was there, the Paleogallus plain. So through the majority of the occupation of these sites that are of World Heritage status, there was a big plain in front of them. So that demonstrates how significant that plain is for understanding these sites that are crucial to understanding the origins of human culture. I just want to pose the research question, why is this early behavioral and cultural complexity on the South African coast, right? Why does it occur first here? And why is it so abundant and spread through time the way it is? And we have to then ask the question, what causes behavioral and cultural complexity in hunter-gatherer societies? So at this point, I'm going to drag you in the direction of anthropology, because we have to go to anthropology to understand the answer to that. And quickly, we have to also ask the question, since you're not anthropologists, most of you, what do we mean by behavioral and cultural complexity? Well, it has a technological realm. How many parts are there to a tool, and how many raw materials make up that tool? It has a sociality realm. How much economic and political differentiation is there in this hunter-gatherer societies? And it also has an institutional realm. How many rules, regulations, laws, and norms are there that exist in that hunter-gatherer society? And all these things vary among hunter-gatherer groups. And I'm going to kind of really quickly summarize what we know about that, and we actually know quite a bit. And that comes from a general, what we call macroecological adaptive system theory. And so this is a body of theory that exists in anthropology that most of you probably aren't familiar with. And it started about 40 years ago, the development of this theory in the social sciences. It's drawn from comparative ethnography, so you take the ethnographic record, so observations of hunter-gatherers, and you summarize it both qualitatively and quantitatively. It comes from human macroecological studies that were initiated by these two people, Oswalt and Binford. And then in 2016, I wrote a paper where I summarized this body of theory and contextualized the South African record in regards to that body of theory. So I'm not going to go through all the bloody details of that, but I'm just going to summarize some of it, and I think you'll get the point. So macroecology, developed by this super famous paper published back in 1989 in Science called Macroecology, the Division of Food and Space Among Species. And in this paper, the authors argue that we should start looking for structural parameters that drive the distribution of plants and animals. So we can do things like plot body mass versus the area and so on. But believe it or not, despite the fact that humans are really weird, right, and strange and hard to predict, humans actually follow the principles of macroecology quite well. So here is a plot of the contribution of the diet from hunting, so how much meat is in your diet. And here's the size of the territory, and each one of these dots is a tribal group. It's an ethno-linguistic group. And you can see that the more meat that's in the diet, the bigger the territory that ethno-linguistic group requires. It's kind of like, so you can think of an ethno-linguistic group in the sense of as a species, right? So there are structural parameters that drive the variation of hunter-gatherer society. And over 40 years of research in anthropology, we've learned a lot about that.
Okay, fantastic.
So
I'll let you know
now what I'm going to do is just summarize two extremes for you that we've learned from this theoretical, this application of theory to data. And here's two extremes of complexity. Here's the Khoisan hunter-gatherers here in the southern African subregion. And here's a polar Inuit from the Arctic. And they're both hunter-gatherers, But they have enormous differences in those three realms of complexity that I talked about, technology and institutions and so on. And just look at the complexity of that Inuit kit. It's incredible. And he's got those very sexy sunglasses, right? Kind of like Geordie LaForge in Star Trek. But the thing that I want to point out is to make those sunglasses, it's about 2 weeks of labor, and they constantly have to be fixed. And there's nothing like that in terms of technological complexity that you see with hunter-gatherers in the tropics. Everything they have can be made in probably 10 or 15 minutes and carried on the back of a person. Okay? So these are two wildly different types of complexity. And so just to summarize what we know is among tropical terrestrial hunter-gatherers and temperate aquatic hunter-gatherers, when the diet is rich in plants and animals, you have very little storage. It's very light that can be carried by one person. The camp can be moved many times a year, 10 to 50 times a year. So that's what we call a residential move, when you move your whole economic group. Territories are large. Sociality is egalitarian. So in other words, everybody's equal in society, so the institutional complexity is quite low. Warfare levels are very low. But when people expand their diet and add aquatic resources to it, you get lots of storage, very heavy technology that becomes hard to move, few, sometimes no residential moves. People often live in villages. Band sizes are quite large, 100 individuals or more, so these are villages and these are small residential groups. Small territory sizes, non-egalitarian, so you start to get chiefs, slavery, all kinds of institutional complexity, and warfare levels get very, very high among these societies. So this is two ends of the extreme, and one of the key additions that creates all this complexity is the addition of those aquatic resources to the diet. Okay? So, and we understand why this is. We have theory that helps us understand why this is. So here's the famous theory of economic defendability that was developed by Jerome Brown back in 1964 to explain why some birds are warlike in the sense that they design a territory and defend it against other birds, and some are not. And what that theory predicts, and this has been looked at among about 240 different species of animals, is that when resources are really predictable and really dense, we see high levels of territoriality or warfare, inter-individual conflict. And what we find in the anthropological record is when we add aquatic resources to the food base, those are highly dense and predictable resources, and the warfare levels go way, way up. So that's just an example that we understand why these hunter-gatherer societies have greater levels of technology, they're more warlike, and so on. We have good theory that fits well with our 40 years of empirical observations. So you probably can see where this goes. So what creates a complex aquatic hunter-gatherer? The addition of aquatic foods adds dense and predictable resources that allows for a reduction of mobility, stimulates conflict, evens out the availability of protein through the year, so it contributes to a high-quality diet, and people can divert time normally devoted to hunting and other activities, foraging along the landscape, to things like social behavior and complex technologies. So an Inuit can sit for 2 weeks and make those really wild sunglasses because they're sitting at one spot because they've generated a lot of resource storage that they can use to reduce their mobility systems. Okay? And you also get population rise when you add these aquatic resources. So, but what we find is aquatic resources are not enough, and we have really good examples. So, for example, California Indians. They have aquatic foods that have been added, but they also add acorns, which produce a productive, storable, nutritious diet, and they have deer in their diet. So my point is, seafood is not enough. Seafood's not enough to create that complexity. It's a key part of the equation, but you have to have another resource base, and our comparative ethnographic record shows that that normally comes from a rich terrestrial food base. So we've had great people, and you've seen them today, doing the studies on the food availability and its productivity. Jan talked about shellfish foraging returns, and what he showed is that when you expand your diet to the intertidal zone, there are times of the month during low spring tides where you get really, really high return rates, but the rest of the month they're quite low. Suzanne showed us that carbohydrates can provide a steady food return, but it's quite low return in most of the habitats. So from what we can see is that if you have just the FYNBOS or the Cape Flora plant foods plus aquatic foods, that's probably not enough to generate the complexity that we see in this South African record. And then Chris Brook just showed us that large mammals on the Paleogallus plain, these very large populations of grazing species, are a super rich, abundant resource, but they're only available when we have glacial periods. So on the south coast, in the Holocene, we have coastal foods and terrestrial foods. But together, they result in a very difficult foraging system. And I think if you run populations into those systems for thousands of years, with the natural tendency of human populations to rise, you have probably an unsustainable adaptation. But when the world swings into a glacial climate, that Paleogallus plain is exposed. We now add a third resource to this resource space here, which is our large grazing animals. And when we do that, we create the ideal conditions for producing a very complex hunter-gatherer adaptation. And that complex hunter-gatherer adaptation is what produces that super rich material culture that is only represented here in the Cape in South Africa at 100,000 years ago. And that's why we have a record that produces something that merits and warrants a World Heritage Site here on the South African coast. So thank you.
Excellent. Thank you so much, Curtis. Yeah, we have some time for discussion, so I don't know if
Sure. Does anybody have any questions?
Questions for any of our speakers who didn't get a chance to answer questions? Fernando.
Thank you guys. I have a question for Richard Cowling. You said that this limestone FYNBOS, part of the richness is explained by the exposure of the Paleogallus plain, and you have shown us this large area of calcarenite that must host also this limestone FYNBOS. But what I have seen is that between that large patch and the present limestone FYNBOS, there is a wide belt of other vegetation that I don't know whether the limestone plant can kind of stepping stone, whether there is communication or not between those patches.
Can you hear me?
Yeah. Can I ask that you use this just for the online guys? Thank you, Richard.
Sure, very good question. Very good question, Fernando. And I've thought about it. You know, you can invoke dispersal, but it's not going to work for a lot of those dispersed species because that intervening band was largely really fertile soils that probably supported grasslands in the east and Renosterveld in the west. But there were all these areas of dunes, and I suspect some of those dunes also had bits of calcarenite in them, and that provided the kind of stepping stone. I don't know, Hayley, whether you can comment on that, but you did say that some of those old dunes offshore were cemented to a certain extent. Yeah. Yeah, it's because of the cemented bits. So those provided probably the stepping stones for the calcarenite flora. From the big bulk of it in the extreme south, to what's left over in the existing CFR. Make sense to you?
Yeah, absolutely. Thank you. Yeah. I actually also have a question to Richard. I was wondering the bits of limestone fynbos and strandfeld that we see nowadays, right? They've been the long-term stable refuge here, right? Whereas the Paleo-Gallus plain was there for a long time, but then there are also times when it blinked out of existence. So what was the role of this Paleo-Gallus plain in driving diversification in those limestone fynbos and strandfeld elements? Was it, did the diversification nevertheless happen in these stable environments that are today's remnants, or was speciation really sped up in those times when this Gallus plain occurred? I mean, are there ways of, with data phylogenies looking at whether ranges of endemics are disjunct or limited? Are there ways of inferring that?
I don't know. I would hope so. I would ask Tony or people like that. But what I do know is that the current flora, certainly the dune flora, I've looked less at the calcarenite flora, is supersaturated. I mean, the incidence of rare plants is phenomenal. And you pick up all these disjunctions. So, there's a weird Thamnocortis that pops up just outside Port Elizabeth that's closely related to the thatch species way down in towards Cape Agulhas. The other disjunctions, so you get little Physcineas and stuff on the calcarenites in the Sundays River Basin, which only pop up again at De Hoop. You get weird remnants of, I think they're remnants, but it could be my bias, of populations of species that, you got to wonder, how could such small areas support so much rarity? So there would be ways, I think, of modeling rarity in terms of area, looking at constraints on population sizes to explore those questions further. But we haven't really been there yet. That's why I think it's a fruitful area for future research, because we understand the dynamic and we're looking at these little remnants.
So I've got a question actually two questions for Hayley. So first, let me start by saying it's really an impressive program in scope and well done. It's amazing, the whole project that we've seen. But my questions really are, first of all, I understand that the sort of survey of the seabed will reveal the texture of the seabed. What's the evidence or the data that underpins the actual reconstruction of the geologies? And how much confidence do you have in the geological mapping? That's the one question, and then I'll ask you the second one after.
Thanks, Tony. Yeah, that's a good question. I didn't go into too much detail about all that because, of course, with 12 minutes, I didn't think that I would do that. But actually the most informative data type is sub-bottom profiles. It's like a high-frequency version of seismics. So there's a slight penetration into the seafloor, depending on the bottom type. If there's bedrock, it stops at bedrock. But if normally if there's any density difference between these layers, almost like slicing through the cake, if that was the seafloor, where there's a density contrast, we can see that. And it's those data sets that allow you to choose good locations for coring, because you can see through the modern marine sand, which wouldn't have been there during the last glacial maximum. It's a very mobile, topmost unit. So although where rocks are exposed on the seafloor, things like multi-beam bathymetry and side-scan sonar are excellent, where there's a layer of marine sand, then they don't do so well. So for most of our map production, I used sub-bottom profiles that were quite widely spaced. And I traced different, they're called seismic facies, according to their textures and according to where we record them, and knew what those represented. So it's mostly based on that. There was, I would say, where we had actual data, I'm very confident with what we've been able to show. Of course, in between those areas, I relied a lot on legacy data to see where we might find similar patterns and how extensive might these things be. So until the whole area is completely mapped, it'll never be, I can't be totally confident. But I'm reasonably confident that we've got a pretty good handle on what was out there 20,000 years ago. Because through all of that, I stripped off the modern sand, looked at where there were cores of aeolianite, so where there would have been bigger dune fields. I looked at where there were changes in the bathymetry from the sub-bottom profiles, but not from the modern bathymetry. So it was all about getting back to what it might have looked like before the sea level rose the last time around.
Thanks very much. The second thing I wanted to ask you, I was actually quite surprised by, well, I'm quite surprised by the sort of two bands of calcarenite. I suppose, I mean, I had an expectation that there would have been connections offshore between the little islands that we see today in the present. But I'm just trying to understand, in terms of the formation of the calcarenites, why you end up with two bands. Is it the case that the regressions and the transgressions are quite rapid relative to the high stand and the low stand periods? So that, do you know what I'm getting at? Because
Yeah,
the
I think
coast would have
so.
been, yeah.
And I think that there are two bathymetric terraces on the Cape South Coast shelf. One of them from 40 meters to about 47 meters, and the other one from about 70 to 75 meters. So the inner shelf extends out to 40, and then there's a little drop. And then the middle shelf extends out to 70, and then there's a little drop. And then there's the rest of the Agulhas Bank. And the Agulhas Bank is obviously at its edge where the continental shelf drops into the continental margin. That's deeper than the last glacial maximum shoreline. So there's a whole lot more out there, all the way out to where the inflection point sits. And it seems as though a lot of the dune material that's now sort of cemented into rock is accumulated up against those two terraces, which I think makes sense geologically. So that probably, and I see a lot of evidence for those things being preserved against that drop-off, those little drop-offs at the two terraces. So I think that's how to explain that. I mean, of course, you have mobile dune material on top of those plateaus as well, but right now it's not very well preserved. So we didn't want to guess, really, that it would have been there.
Sure, I guess if you had intervening islands, it could start to explain better some of these, or Richard's story around the migration. Anyway, thanks a lot.
Thanks.
So my question is linked to that little study that was done on the CO2 and the impacts of low CO2 on bulb size and productivity. And I was just interested to know whether or not that's been thought about for just more generally the feedbacks to vegetation as a whole, for example, and how that might have impacted large herbivores and fire regimes and other things like that could ultimately also influence your modeling of the paleoscape into the past. I'm not sure who wants to...
Oh, there's another one coming at me. Well, there isn't really that much you know, there's like 5 trillion bits of experimental evidence for what happens to metabolism and the different compartmentalization of photosynthates under high CO2 experiments. But, you know, your experiment under low CO2 is relatively rare. Not that much interest in it, really. So the literature seems to be small about what happens to plants under low CO2. But as far as we can tell, so the studies that have been done seem to establish that the trends from LGM to pre-industrial to future tend to be like approximately a straight line, or maybe they curve, but at least it's monotonic. So so there's a lot of studies that compare what happens to plants, nutrition for people, or I guess by extension to ungulates or something, under pre-industrial versus future anticipated high CO2, with these kind of classic papers about threats to human nutrition because grains will be less nutritious. So the general pattern that emerges is that as you increase the CO2, you get higher carbohydrate and sugars, but less nitrogen and minerals. So we really don't have, as far as I can tell looking at the literature, we really can't confirm that experimentally for LGM CO2, but if you do extrapolate that, it suggests, and your oxalis study at least confirms the less carbohydrate and sugars, it seems, and everything is smaller under low CO2, but the implication is that nitrogen and mineral content would be higher. So I haven't thought about that from the grazer point of view, but from a human point of view, it implies that pre-industrial geophytes are maybe analogous to potatoes, but in the LGM they're more like beans. So that's obviously a big, profound difference in food quality, but it's not better, or I don't know whether it's better or worse for a human's diet. It's less calories, but they're much more, you know, there's less energy. I don't know how they got enough energy, except by eating shellfish fat, I guess. But yeah, so the plants are more nutrition packed, more energy. It's more nutrition dense food, but there's clearly is a shortage of carbohydrates from plant foods. So that would presumably also be true for animals, but you would also expect all that vegetation to be less flammable. So other studies of what happens to LGM vegetation in general is that like sclerophyll is reduced. So sclerophyll is expected to increase in high CO2 because it's just all this free photosynthate. It's very easy to produce photosynthate, so you can just load it into your leaves. And so it seems like the emerging view of what happens to plants at the LGM, well, there's actually a lot of unexpected things that happen to them. So there's that study that was done on those junipers at La Brea or somewhere in California, at the LGM, where they looked at the carbon isotopes of growth rings and measured the thickness of growth rings and came to the conclusion, that was published a few years ago in Scientific Reports, that growth rate didn't really change. Growth rate was actually, I think, constant, but because of the carbon isotopes, they came to the conclusion that these plants were close to carbon starvation at LGM CO2 levels. And so the only way they can make that budget balance is to say that plants put less resources into roots. And that's only possible, probably, if you're a tree, these were junipers, living in a place that's wetter than today, which is already independently reconstructed for the southwest US, that it is one of the places on Earth that's wetter than today at the LGM because of the shifts of the jet stream. So if you're a tree, you're barely making a living, but you don't have to do that many roots, apparently, because it's so wet. I don't know. That hasn't been really replicated elsewhere, that kind of study of tree rings and carbon isotopes, but it's quite an unfamiliar world, the LGM. Yeah. If that answers your question.
Thanks. Thank you, Kel, for that answer. Any other questions, comments? Nothing. No. All right. Thank you very much to our speakers. Yeah. Again, I'm biased, but I enjoyed all your talks. So thank you very much for that.
The efficiencies that we derive from collaboration in the study were enormous. Together from different fields, they work collaboratively without animosity, and I think that's because we were able to achieve so much in such a little time.
Agreed, completely, yeah. It's a great bunch of people here. So, thanks, Richard. All right. Yeah, we're a little bit behind time, so we've shifted the keynotes, I think, to 10 to 1. So we'll take a little break now, and then 10 to 1 we'll come back for the keynotes. I think there might be 2 parallel keynotes, if I understand it correctly. Yeah. Thanks. No. I don't think we're opening up the rooms. I think we're just going to all get into one. I'm not sure which one it is in, but we'll just see where everybody goes. Thank you, everyone. started.
Thank you so much for having me. Today I want to talk about some of my work with students and colleagues on native California plants on serpentine soil. So, as we probably all know, Mediterranean climate ecosystems host the most diverse extratropical floras. From the past couple decades of research, we know that this has been promoted by long-term climatic stability compared to surrounding areas that has led to generally low extinction rates. So, Mediterranean climate ecosystems host both young and old species radiations. They're also marked by high levels of endemism, not just to those geographic regions, but also habitat endemism. For plants, this often takes the form of edaphic endemism, in which plant taxa are restricted to specific soils, especially nutrient-poor or otherwise harsh soils. In South Africa, that might take the form of restriction to limestone or quartz. In California, serpentine soil plays a big role in levels of endemism. So although serpentine soil only covers about 1.5% of the state's area, it hosts about 13% of the endemic species. And there are nearly 700 serpentine-associated taxa. So serpentine habitats are an important contributor to the diversity of our flora. And what is serpentine? It's an ultramafic rock from the Earth's mantle that rises to the surface at tectonic plate boundaries. It often has this very cool green or grayish color to it. And once serpentine comes to the surface, it begins to weather and gradually accumulate organic matter. So there's a lot of variation in serpentine soil, especially in things like rock size, water holding capacity, nutrients, and the specific heavy metals that are present. But there are also some commonalities. The soils tend to be very high in magnesium and very low in calcium. This is tricky for plants because magnesium and calcium ions can be similarly perceived and uptaken by roots, but plants need a fair amount of calcium and very little magnesium. Serpentine is particularly abundant in California, especially in the Sierra Nevada and coast mountain ranges. And because serpentine tends to occur in discrete habitat patches, it can be a source of strong divergent selection. Also, adaptation to serpentine has occurred many, many times independently. In the California flora, it shows up in about 50 different plant families. So this means that we have many evolutionary replicates of serpentine tolerance and serpentine endemism, and we can use that to look for common patterns and general principles about how plants adapt and specialize on different habitats. Before I go any further, I want to give you an outline of a couple questions I focused on in my research program that I want to touch on in this talk. The first is, does adaptation to serpentine soils cause reproductive isolation? And the second is, why are some serpentine adapted plants restricted or endemic to serpentine, whereas others can grow both on and off serpentine? Starting with the first question, there are two major mechanisms hypothesized. First, if there's selection against migrants between edaphic habitats, that can keep populations or species from interbreeding. This is best examined with a reciprocal transplant experiment in which both taxa are planted into both soil types and we get some measure of fitness. And what we expect it to see is this crossing fitness function where the plants have a home site advantage in their local soil. Second, divergence in flowering time may come along with adaptation to different soil habitat, so that populations or species are reproducing at different times and hence reproductively isolated from each other. So early in my career, I started investigating the role of serpentine adaptation in reproductive isolation, and I worked on 3 different systems that essentially represent different points along a speciation continuum, from populations to fully isolated lineages. For each system, we characterized the different habitats, we used reciprocal transplants to understand selection against migrants, we quantified flowering time divergence, and we used molecular markers to estimate gene flow. I'm just going to briefly review this work for you as a way to set up some of my more recent work that I want to talk about in more detail. The first example is Leptosiphon parviflorus, which has adjacent populations both on and off serpentine, associated with a simple Mendelian flower color polymorphism. White flowered plants are unable to survive on serpentine at this site in the coast ranges, but pink flowered plants do just fine. They also have genetically based differences in flowering time, but when we use neutral genetic markers to look for genetic divergence between these populations, we didn't find any differences. So they're experiencing plenty of gene flow. The next system I want to touch on is the coyote mints, the genus Monardella in the Lamiaceae. This is in the northern Sierras, and there are two species there that are both strictly endemic to serpentine, but they occupy slightly different serpentine habitats, as shown here on this PCA of various aspects of their habitat. They each have a strong home site advantage in a transplant, and they flower at different times, even though they share pollinators. However, they do co-occur on one big serpentine outcrop with intermediate habitat, here indicated with the red arrow. And at this site, they co-flower and experience hybridization, and that leads to integration into each species away from the hybrid zone. So these interfertile species have a moderate amount of integration that's facilitated by some intermediate habitat. Finally, I worked on two cryptic species of Lasthenia in the Asteraceae that co-occur on serpentine outcrops in the coast ranges. Although they're morphologically very hard to distinguish, they don't hybridize. Our field study showed strong home site advantage in reciprocal transplants and genetically based divergence in flowering time that allows them to coexist. So what can we say? We've got three different systems, closely related populations in Leptosiphon, hybridizing young species in Monardella, and coexisting but not hybridizing species in Lasthenia. And we can, but we see when we look at the genetic data that they actually have, so we see that they actually have similar degrees of flowering time isolation, and they have similar strength of selection against migrants. But when we look at the genetic data, that seems to be unrelated to the strength of reproductive isolation as measured by these two components. So it seems that if the reproductive isolation or the genetic divergence is already in place, then the edaphic divergence might help maintain it, but it doesn't actually get it started or prevent hybridization. So is this a general result? The problem here is that we don't know whether this is just anecdotal or if it's really a consistent pattern. What we'd like to do is replicate across these degrees of evolutionary divergence and ask how divergent adaptation to serpentine contributes to reproductive isolation or limiting gene flow. So that brings me to the next study, which was led by my former PhD student, Shelley Sayanta, who's now a postdoc at University of Minnesota. And what we decided to do was use the many times that California plants have independently adapted to serpentine as evolutionary replicates to try to find general patterns. So we picked out many taxon pairs at the population level. These are populations of the same species on versus off serpentine soil, and we'll call these tolerator pairs. And we picked out many taxon pairs at the species level. These are a serpentine endemic species paired with its sister species not on serpentine. And what we call these endemic pairs. So we ended up selecting 17 sister taxa pairs that span this speciation continuum. We had 9 pairs of tolerator sister taxa. Here's an example from Clarkia. And we had 8 pairs of endemic sister taxa, an example here of Colomia. So overall of these 17 taxa pairs, now represented here by a photo of the serpentine member of each pair, They spanned 9 genera and 6 plant families. And to be able to compare similar life forms, we did limit the study to annual herbs. First, I want to tell you about our results from this study on flowering time divergence for sister taxa on versus off serpentine. So the questions we asked here were, first of all, does edaphic divergence predictably result in flowering time divergence? And is flowering time divergence predictably greater in taxa pairs further along the speciation continuum, so the endemic pairs? And we did this by setting up a greenhouse reciprocal transplant experiment where for each of the 17 sister taxa pairs, we went out into the field and collected local seed and local soil. And then we reciprocally transplanted each seed source into its home soil and into its sister taxon soil. And this design allowed us to estimate how much flowering time divergence each pair had and how much of that divergence was genetic versus a plastic response to the soil. And of course, we replicated each taxon multiple times with many maternal families. We quantified the days to first flower, and we did biweekly censuses to calculate the overall flowering time distributions so we could look at their overlap. First, I want to show you a summary of the results for days to first flower across all pairs. So in this graph, tolerator pairs will be in orange and endemic pairs in blue. And if the members of a pair have no flowering time divergence, they will, that point will fall along that 0 horizontal line. Whereas above the line means that the serpentine member flowers later, and below means that it flowers earlier. So you can see clearly that the flowering time divergence is common, and most often the serpentine plants flower later, perhaps because they need more time to reach the size of flowering on nutrient poor soil. The average shift was about 19 days. And when we examined it by pair type, we see that the flowering time divergence wasn't any greater for endemic pairs. We also broke this down into genetic and versus plastic responses, and I'm not showing you that data here, but we didn't see any difference between the portion of the variance that was genetic versus plastic between endemic and tolerator pairs either. So the take-homes from this study were that adaptation to serpentine predictably results in genetically based flowering time divergence, but it doesn't differ between different points on the speciation continuum. I should also note that in most cases, flowering time still overlapped, which would allow some cross-pollination if populations are nearby. So what can we say after this big comparative study? We'd have to conclude that serpentine adaptation can contribute to speciation as defined by reproductive isolation, but not necessarily cause it. So my guess is that there needs to be at least some geographic isolation to really get things started down the speciation path. So moving on to that second question, why are some serpentine adapted plants restricted to serpentine, whereas others can grow both on and off serpentine? So one hypothesis is that there might be strong costs to serpentine adaptation that promote endemism. And you might be wondering why, if all these plants are adapting to the same soil type, there would be different costs associated with serpentine adaptation, but there's actually a lot of variation in serpentine habitats. And it might be that adaptation to, for example, rocky serpentine barrens will come with bigger fitness trade-offs than adaptation to serpentine grasslands or serpentine seeps, and that endemics might be more likely to occur in these harsher habitats. So again, with Shelley Sayanta, we tested this idea by quantifying soil chemistry in a serpentine population from the 8 endemic and 9 tolerator serpentine taxa that I already told you about. And what we found is that the only significant difference between the types of species was that endemics occurred in serpentine soils with less calcium than tolerators. And this is a really interesting result because calcium is the sort of quintessential nutrient deficiency of serpentine soils and has been found from nutrient addition studies to be the primary limiting nutrient in serpentine. Now, you may be wondering what those costs actually are. Why would serpentine adaptation lead to low fitness in non-serpentine habitats? And this question was addressed by some classic experiments by Art Kuckerberg in the 1950s. In these experiments, he took serpentine and non-serpentine populations of a tolerator species, in this case common yarrow, and reciprocally transplanted them in each other's soils. He found that in serpentine soils, individuals from non-serpentine populations couldn't survive. But in the non-serpentine soil, he found that both plant types grew just fine, suggesting that adaptation to serpentine didn't come with a fitness cost of growing in non-serpentine soil per se. Instead, Art proposed that it was competition in non-serpentine habitats that limited serpentine plants from growing there. So if we extend this hypothesis to the evolution of tolerators versus endemics, we would imagine that when serpentine adaptation comes with a larger loss in competitive ability, it would lead to endemism. So again, as part of this study with Shelley, we used the same suite of 9 tolerator and 8 endemic species to ask whether endemics were poorer competitors than tolerators. So in this experiment that I already told you about, we actually added an additional treatment of competition to the non-serpentine soil. So we grew a standard competitor, which was a native California grass, Bromus carinatus, in half of the non-serpentine pots. And what we expected is cartooned here. So we're taking the serpentine plants and planting them into non-serpentine soil with and without competition. And we hypothesized that the endemics should show a bigger decline in fitness with the addition of competition compared to tolerators. To measure the competitive ability, we quantified log response ratios in fitness, which is sort of a standard metric of competitive ability. And what I'm going to show you on this graph is the pair type or the taxon type on the x-axis, the tolerators versus endemics, and the log response ratio on the y-axis. If there's no effect of the grass competitor on fitness, then taxon is going to fall right along that horizontal 0 line, whereas better competitors will have higher values and worse competitors will have lower values. And what we find is that indeed, endemic species have lower competitive abilities than tolerator species. This is an exciting result because it supports Art Kuckerberg's initial hypothesis, which had been upheld in a few taxa here and there, but this is sort of a comprehensive comparative result. And it suggests that serpentine endemics may be currently restricted to serpentine habitats by competition. So we can also ask, why are endemics poorer competitors than tolerators? And there's a couple hypotheses that we came up with to think about this. The first one is that they might have evolved from non-serpentine ancestors that were already poor competitors. And we call this the pre-adaptation hypothesis. Alternatively, it might be that the adaptation to serpentine in these endemic lineages caused them to become worse competitors. And this is what we call the trade-off hypothesis. And so as part of that experiment, we also grew the sister taxa to each serpentine taxon so that we could quantify its competitive ability as well. And then we calculated the divergence in competitive ability for each pair. So the results I'm going to show you have the pair type on the x-axis, tolerator versus endemic, and the divergence in competitive ability on the y-axis. And what we see is that endemic pairs have undergone more divergence in competitive ability than tolerator pairs, which have undergone, on average, none. And this really supports that trade-off hypothesis that endemic species have adapted to serpentine, and that adaptation has involved more trade-offs in competitive ability than tolerator populations. So the take-homes from this are that endemics are found on soils with lower calcium than tolerators. Endemics have lower competitive abilities than tolerators. We see this as the cost of serpentine adaptation. And endemics have actually lost more competitive ability while adapting to serpentine. So do we have answers from all of these different studies? First of all, I think we can say our data show that adaptation to serpentine seems to contribute to maintaining species, but not necessarily cause speciation. And endemics are found on harsher soil. They have lower competitive ability, and they've diverged more in competitive ability. And this really fits in with comparative data across Mediterranean climate ecosystems, showing that regions with nutrient-poor soils tend to have higher endemism and diversity. So with that, I would like to say thank you to my mentors and collaborators and students and greenhouse managers that made all of this research possible, and to you for listening. And I'm happy to take any questions that you might have.
Thank you so much for a fascinating talk on that edaphic endemism. Do we have any questions?
Thanks. Hi, Kathleen. This is Hugh Safford. It was nice to see you on the screen.
Oh, hi, Hugh. Thanks for coming.
Yeah, you're welcome. That was great. Hey, so I have a question for you. I mean, given what you're finding, how well do you think your findings would extend to tropical ultramafic habitats where things like cobalt and chromium are actually bioavailable? Sorry, that's a hard one, I know. But I want to know.
Yeah, I can't really think of a reason why they wouldn't apply necessarily. Address that.
Interest is, hold on a second. So you can hear me? Yeah, I think my interest is because I think that in that case, I'm just wondering whether or not the decline created by the soil toxicity might be so much stronger because you've got that in the availability of the heavy metals in the system, whether or not it might actually be driving speciation. You know what I mean?
Mm-hmm.
Anyway, just maybe something for you to get another student going on.
I think that the fitness costs or the selection against the non-serpentine taxa on serpentine, even in the temperate, is very, very strong. I mean, almost everything dies if you put the non-serpentine taxa on serpentine. So I don't think it's really an issue of the fitness cost not being strong enough. But I could imagine that more toxic heavy metals could also lead to slower growth, which I think is what contributes to the lack of competitive ability. So I guess I don't see that being different, but it would be fun to work on.
Thanks for a great talk, Kathleen. It went a bit fast, but I'm trying to remember the competition experiments. Were they conducted on normal soil only?
So they were conducted on the non-serpentine soil that was collected in the field from the non-serpentine member of the taxa pair. So it wasn't the same soil across the entire experiment because each pair that we're using as sort of an independent evolutionary replicate had its home non-serpentine soil.
Okay, so yeah, that answers it. But so to conclude that the serpentine species are poor competitors, I wouldn't agree because they're better competitors on the serpentine soil, aren't they? So competition is context dependent, and you can win in some environments and lose in others. And the serpentine species are winning on the serpentine. and the other species pair is winning on the non-serpentine soil, the other species from the pair.
Yeah, I guess it depends like how narrowly you want to define competition, because we did actually measure competition on the serpentine soil because there were too few individuals from the non-serpentine taxa that lived. So it's more of a inability to survive at all, regardless of whether there's a competitor. So I don't know if you would call that competition.
Yeah, I'm not sure.
But I get what you're saying.
Yeah, it's just that competition is context dependent. So to call something a superior competitor, well, in which environment? That's my question, I suppose. So, yeah.
Right. So I guess if we're asking what limits the serpentine taxa to the serpentine, what prevents them from expanding off serpentine, then we want to ask what happens with competition off serpentine.
Yeah, and what limits the non-serpentine species to non-serpentine soils? Well, it's their inability to be superior competitors on the serpentine.
Well, I think it's the inability to survive in most cases, regardless of whether there's a competitor or not.
So if you grow them in pots without a competitor, they just don't make it even at all? Okay, so it's that harsh, right? Okay.
I mean, there are some tolerator species that you can take the non-serpentine populations and plant them on serpentine and they will survive because, you know, they're harboring tolerance alleles. And that just wasn't the case for the pairs that we happened to include in this study. We didn't choose them based on, you know, any pre-knowledge of that, but the ones that we happened to select, the non-serpentine ones, were generally all dead on serpentine, or maybe, you know, one or two replicates would survive, but not enough to estimate any competitive ability.
Okay, thank you very much.
Thanks, y'all. Let's take one or two
Thank
more
you.
questions.
Hi, thanks for the talk. This is John Williams from UC Davis. I was just curious that you found that there was the serpentine plants had delayed flowering, and I was wondering if you think maybe could that potentially just in the Common Garden experiment. But I worked on plants in the Shasta Trinity, and those hot serpentine patches seemed, anecdotally anyway, the flowers were very early in their phenology, and because they got fried, it was so much hotter. And I just wondered, any thoughts on that?
Yeah, actually that result surprised me because there are some really classic examples of serpentine plants flowering earlier than their, you know, close relatives off serpentine. There's, you know, monkey flowers that are known to do that. And, you know, there's a few anecdotal cases. So we were expecting that because serpentine soils often dry out faster. Like you said, they get really hot and they're exposed. I think that's not consistently true across all serpentine soils. So there are a lot of serpentine soils that, you know, are quite wet and there's serpentine seeps, and there's also a lot of non-serpentine soil that we used in this study that it's not great soil. You know, it's not like comparing it to potting soil that is very cush. So yeah, I think we did have a few taxa pair that showed that pattern of early flowering in serpentine, but that was the exception, not the norm. The norm was that the serpentine ones grew quite slowly and just seemed to take longer to get enough biomass to be able to make a flower too.
Thank you.
I should also say that we did look at the phenology of those plants in their native habitats, and it showed that same pattern. So it wasn't just a greenhouse effect.
Let's take one more question. I think Fernando had his hand up for a couple of rounds there, so let's go to him.
It's going to be quick. Thank you, Kathleen. This is Fernando Ojeda from Spain. My question is that is there anything going on below ground? I mean, microsymbionts. Is it?
Probably, yes, but we did not include that in the study. It just didn't seem logistically feasible to do, multiple transplants across this many taxa, and also work on manipulating the microbial community. But I do think that could be potentially very important, and it's something we just left out. And so that's definitely a weakness of this work. We sampled the field soil, but we didn't do any kind of treatments to get rid of the biota.
Yeah, I would say it's worth it to look at that, to look at the microbial flora in the soil. Anyhow, thank you very much.
Thank you.
All right, thank you so much. Yeah, we're gonna end our questions there. Yeah, okay. I just want to thank you so much for coming online and for preparing the talk for us and for sticking around during our delay. We really do appreciate it. So it was definitely
no worries. Thank you so much for having me.
Thank you. Can we give a round of applause, everyone? Okay. I think from there we're going to go straight into our next keynote address by Alexandra Seifert.
Hello. Thank you so much to the conference organizers for inviting me to be a keynote speaker at Medecos. I'm really sorry that I'm not there to see everybody in person, so hopefully we have another chance in 4 years at the next Medecos. My co-author is John Keeley, and I would like to share with you some of the lessons we've learned in complexity studying the impacts and effects of global change on wildfire in California. I've been studying fire for a long time, more than 2 decades, and I've noticed that in recent years, more than ever before, wildfire has become a really hot topic. It seems to be in the news all year now, whereas it used to just be in the news during fire season. And part of this is because it's become a possibility all year in many different areas. This picture on the right is showing you in the dead of winter, on New Year's Eve this past year, there was a fire that broke out in Boulder, Colorado, a grass fire in which more than 500 houses were destroyed. And of course, it's not just in the United States. There have been major fire events occurring around the world in recent years with really serious human impacts, many lives being lost, many structures destroyed in these fire events. And of course, there's fire events going on just this summer with the heat wave in Europe, fires in a lot of the Mediterranean parts of Europe, in Spain, France, Portugal, and wild and urban interface fires in London, which is not to be expected. So with all of this fire occurring and with fire in the media a lot, we keep hearing this phrase, the new normal. Wildfires are roaring across the West again. Even the United Nations released a report associating wildfires and heat waves with a new normal. But the question is, especially for the person who's not a fire scientist, the question might be, well, what is the old normal then? And what is new normal? And why is it new in terms of fire? And of course, that's a really complicated question, and it's hard to answer because wildfire is not only a hot topic, it's a really big topic. Just in fire management and fire ecology, there are completely different languages and vocabulary, different methodologies, and fire also includes a wide range of other disciplines, from meteorology through public health. So wildfire is a complex topic, and what I want to do today is, like I said, share some lessons that we've learned in the research that we've been doing for the past couple of decades on altered fire regimes in California, which of course is a Mediterranean climate region. Our first lesson is that there are actually many variations of normal and many variations of new normal in terms of fire. What is normal in fire in California? Well, like other Mediterranean ecosystems, California has tremendous diversity in topography, has tremendous species richness and diversity, different land cover types, different land uses, and these all manifest themselves in a specific signature of fire in different regions. Looking at the historical fire count, you can see there's a distinctive spatial pattern. In some areas, fire burns repeatedly, while in the same time frame, other areas haven't burned at all. And we used a clustering methodology, actually, and we're able to define 8 distinctive fire regime ecoregions in California. But there are 2 broad distinctions in fire that have been causing a lot of confusion. And these basically have to do with, 1, the dry mixed conifer forests of California. These are higher elevation, typically ponderosa pine forests that historically experienced frequent fire return intervals every 10 to 20 years, typically low to mixed intensity fire, often understory, sometimes crown. These are the dark green areas you see on the map. And conifer forest actually occupies only about 20% of the state in California. Much of the rest of it is in shrublands and grasslands. And so the other big fire regime classification that we have is in the chaparral shrublands that have fire rotation or return intervals that have been much longer than those in the forests, with fires returning every 20 to 70 or even longer years. And the natural fire regime for chaparral is actually a large periodic high-intensity crown fire. And what's interesting is there have been completely opposite changes in their fire regimes in the last century or so. This map, produced by Hugh Safford and colleagues, and I know Hugh's been at the conference, is showing you basically the departure of fire return interval relative to the fire return intervals that were there prior to Euro-American settlement. In the blue colors, therefore, we're seeing areas that have burned much less frequency relative to they did pre-settlement. And in the warmer areas are places where fires have been burning a lot more frequently than they did historically. And the blue areas are largely in the conifer forests I was talking about, where there has been very effective fire suppression, and it has excluded fire from much of the forests that typically experienced frequent fire. And as a result, there's been a change because there's been an increase in stand density that is not being burned in the fires anymore. On the other hand, with human ignitions, population increase, urban expansion, frequency of fire is increasing, and it's also, the spatial pattern is changing because as humans move farther into the undeveloped areas, they take ignitions along with them. And so another lesson is that different pathways can lead to similar types of effects. Ecologically speaking, in California, in these different regions, there is a beginning and a progression of vegetation type conversion as a result of altered fire regimes. Vegetation type conversion being a change in life form, whereas forests are now starting to experience a conversion to shrublands, and the shrublands, the chaparral shrublands, are starting to, and actually have been for quite a while, converting to invasive grasses, herbaceous species. And even though there is fire-driven type conversion going on across the state, it is because species are not adapted to fire per se. Species tend to be adapted to a specific fire regime. And the recovery of species and the ability of species to withstand fire or recover from fire is a function of their species traits and the adaptations to the fire regime that they have experienced historically or other disturbances that their traits are appropriately help them be resilient to. In the forests, what has happened is the fuel accumulation that I was talking about has caused a more competitive environment for water resources, and we had a very long severe drought from 2012 to 2015 that in and of itself led to a lot of forest mortality, but then that was coupled with bark beetle outbreaks, and if you can see the picture on the top right, led to very extensive forest mortality in a lot of the forests in the state. And in recent years now, we've been having uncharacteristically severe fire with large patches of high severity fire. And whereas the old trees used to be able to persist through the low intensity fire, they're now experiencing crown fires. And the issue here is that with large patches of high severity fire, the seed sources then become distant and it's difficult to regenerate, especially when there are shrublands that are starting to recover and regenerate where the forests once were. The shrublands, on the other hand, the issue has been that, ironically, the species that require fire for the establishment of new populations, because they have a long-lived, persistent, fire-cued seed bank or serotinous cones, they typically need a period of time in California, 15 or more. We've seen some needing 20 to 25 years to be able to replenish the seed bank or to be able to recover enough so that if another fire comes through, that they can again regenerate. But what's happening is there have been increasingly short interval fires and species are being extirpated. The grass, which is very ubiquitous across California, easily colonizes the disturbed areas. Once it's there, it's highly flammable, so it actually can result in this positive feedback cycle that we call a grass fire cycle. And we have been doing research on this vegetation type conversion in the chaparral of Southern California, where we've looked at the extent of change and the rate of change and the drivers of change over the last 70 or so years. And the map you see on the left shows you in the warm orange and red colors where there has been loss of woody cover and conversion to invasive grasses. So it's been quite extensive. And we've also found that the top drivers vary a little bit from region to region, but short fire return intervals have been the top correlate to the vegetation type change, and soil moisture has also been a concern, and it's been strongly correlated with vegetation type conversion. And I think that has something to do with the ability to recover post-fire, drought fire interactions. And of course, we have different fire regimes that have been altered different ways in different regions, but these different pathways also look the same to humans, because regardless of where you are, the impacts to humans tend to be having structures lost. In California, in just a 2-year period, we lost about 40,000 structures in several fires that resulted, each one with more than 1,000 structures being destroyed. And a lot of people have also lost their lives in recent years in California. Lesson 3. Management effects vary by fire regime or by altered fire regime. One of the most common ways to manage vegetation, or to manage fire, is through vegetation management with mechanical treatments or prescribed fire. And the effects and effectiveness of these treatments vary depending on where you are. So in the conifer forests, from a fire forest resilience perspective, given the increased fuel accumulation, there is a real need to reduce the density of the understory, to use prescribed fire and mechanical thinning to increase the resilience of those forests, and also where there are humans or where there are houses to reduce fire risk to them. So there are mutual benefits for humans in forest resilience in the conifer forests, but in the chaparral shrublands, invariably speaking, vegetation management has a negative ecological effect, partly because it involves removing the canopy because there is no understory in chaparral. And adding more fire to the landscape is adding more fire to an already oversaturated environment of fire. And in terms of effectiveness, during severe fire weather or wind-driven fires, we found that the main role really for protecting structures, which is the objective in Southern California, is to provide a safe place for firefighter access, to give them a place for defensive activities. So they're really best when placed strategically near communities where they can save human assets and human communities. And it's important to understand there are no ecological benefits of this in Southern California. And of course, there are many tools in the toolbox when it comes to fire management and ways to reduce fire risk to humans. And I'm not getting into those in this talk, but an important consideration is that it's important to consider where you are, what the natural ecology of the region is, how fire regimes have been altered, and to match the strategy appropriately to the context and objective that you are trying to achieve. The fourth lesson is drivers of altered fire regimes vary geographically. The first example has to do with climate change. Now, has been a common assumption that climate change is monotonically increasing the risk of fires. And this makes sense because one would expect a more flammable environment with hotter temperatures and drier conditions. But John and I have been looking at fire-climate relationships historically in small subregions across the state of California instead of at large areas like a lot of studies have done. And what we found is variation in the type of climate variables that are significantly associated with fire activity and the strength and significance of those relationships. In the montane conifer forests, we found a significant effect of seasonal climate activity with fire activity in ways that you would imagine, such as increased spring or summer temperature, precipitation, vapor pressure deficit. And in these regions, the story is consistent with what the concerns are, is that climate change is likely to worsen fire conditions. But in the shrublands, interestingly, we did not find any significant relationships between seasonal climate activity and annual fire activity, except for prior year precipitation, which we believe is related to grasslands, because a wet year can increase biomass of herbaceous species. And if there's a dry subsequent year, that means there's a lot more fuel to burn and it's dry. But there's a reason for this, and that is that in Southern California, after 6 months of a summer drought characteristic of Mediterranean ecosystems, the fuel moisture is already very low, and the chaparral is a dense, flammable vegetation type, and the temperature tends to be very hot. You can see the low temperature in the summer and the bottom chart of the South Coast is the same as the top temperature in the Sierra Nevada. And this is not, so basically in Southern California, every year is a bad fire year, climatically speaking. So qualitatively speaking, it can't get a lot worse than it already is. And this isn't to say the climate is not going to have effects on fire in Southern California. It's just more likely to be indirect, such as drought leading to dieback of vegetation that can facilitate windborne embers contributing to fire spread, vegetation change, maybe helping to facilitate vegetation type conversion. But the big concern in Southern California is land use change and human ignitions. And it's also notable that in other studies looking at future projections of the effects of climate on fire regimes, is they found also geographical variability. In some regions, fire or climate is expected to increase fire, but in other regions, it's expected to have a dampening effect on fire. So it depends on the region. And in terms of human impacts, of course, humans do cause most fires. In California, with low lightning ignitions, it's more than 90%. But this is occurring across the United States, and it's also occurring globally, an increasing prevalence or proportion of human-caused ignitions. And John and I recently, with others, have done a study in which we tried to identify the main correlates with area burned and Santa Ana wind-driven fires. A lot of people believe that Santa Ana winds are a precursor to fires, that having more winds means there's going to be more fire. But we actually found that 75% of all Santa Ana wind events do not have a fire associated with them. And that's because there wasn't an ignition during that event. Obviously, there's no fire if you don't have an ignition. And we found that the #1 correlate with area burned in Santa Ana wind fires is the number of ignitions that occurred, in addition to wind speed. This was more than daily weather conditions and more than seasonal climate. And in another study, they found, interestingly, that human-ignited fires actually resulted in more extreme fire behavior and worse ecosystem impacts. And this makes sense if you think about the fact that human-caused ignitions are likely to occur throughout the year. And so that just numerically increases the odds that a human-caused ignition will coincide in space and time with extreme fire weather and result in faster rates of spread and worse fire conditions. And so that is just to say ignition prevention may be something worth considering in the fire management toolbox. And we found that even different human-caused ignitions have different spatial effects on the landscape. And so you can customize your ignition prevention strategies based on the timing and pattern of different sources of ignitions. And the final lesson is that differences in scale or focus of analysis can lead to opposite conclusions. I'm giving you a brief example out of many, but it's related to ignitions, and that a lot of times people think that fires occur close to roads, and that's often because fire ignitions do happen very close to roads, and a lot of different studies have shown that. But if you're considering area burned, the relationship becomes completely opposite, in which larger fires and more area burned actually tend to occur farther from roads. And this makes sense because, of course, in more remote locations, there's more difficult access, there's probably more continuous fuel to burn. But it's just important to consider the context in which you're making decisions. And this is showing you maps of what I just showed you. So this is a model of probability of ignition with the ignition points in the black here. And you can see that when you change that to probability of large fires, the place on the landscape changes, and large fires are likely to occur in completely opposite places of the landscape. Although some ignitions that start in human-dominated areas obviously spread into more remote areas. So to summarize, we had 5 different lessons in complexity, of many, in terms of fire. There's not just a new normal, but there are many variations of normal and new normal. And different pathways can lead to similar effects. So despite this, management efficacy varies by fire regime because there are different normals and new normals. And these are important to consider when making decisions. Also, drivers of ultra-fire regimes vary geographically, and depending on the scale or focus of your analysis, that could result in different or opposite conclusions. And so my overarching lesson, just a silly thing I made up, was mind the gap and know your size. Be careful how you generalize. And I think that's really the message, is that a lot of times there's overgeneralization that can lead to unintended consequences. In terms of dealing with complexity, obviously we need to make generalizations because science is for sharing and learning and taking lessons from one place and applying them to another, just like I am with California, trying to make generalizations to other Mediterranean regions. It's just knowing when and how your conclusions may or may not apply. So when doing research in one region, the objective might be important to consider. So what is the question you're asking? which are the fire characteristics that are important here? Which fire regimes are applicable? How might this mediate the effects of decisions? And to remember, there's more than one tool in the toolbox to deal with these differences. And my ending quote is one, hopefully, that is true, and that is, simplicity does not precede complexity, but follows it. Given that we learn and understand our complexity, it will make decisions much easier in the future. Thank you very much.
Thank you so much. Ooh, bit of feedback there. Thank you so much, Alexandra, for that talk here. We also have had rather catastrophic fires in the past 5 years in the Cape. So, yeah, it's certainly quite a pressing and complex issue across all of our Mediterranean ecosystems. So, yeah, thanks again for joining us live to take some questions.
Thank you. A couple of things. It's Clive McDowell speaking. I didn't hear any mention of, maybe I wasn't concentrating on managed burns, especially in the woodland zones, number one. And number two, the pathogens, the armillarias, the Phytophthora, and of course the borer. Are they enhanced by the climate change, or would they have happened without the increased desiccation, etc.? Those are the 2 points I wanted to clarify, please.
Right. Well, in terms of... Oh, I'm hearing myself echo. In terms of managed burns or prescribed fire, I think that fits into the overall vegetation management that I was speaking of, in that in certain areas in California, there are definite benefits to prescribed fire because it can help clear the understory that has become so dense with shade tolerant species, because fire has been excluded from those forests. And the risk in those forests is that this understory is becoming ladder fuel leading up to the crown, which can lead to crown fires that kill the ecologically important old trees. But in the shrublands, there is an oversaturation of fire. So adding more fire to that landscape is not likely to have any beneficial ecological effect, and it could actually facilitate type conversion to grasses, which become more flammable. In terms of pathogens and insects and climate, that's not really my specialty, so I hesitate to say too much about that, but I believe there is likely a link between changes in fuel, moisture, and forest conditions and pathogens. I just don't want to say anything that's not correct.
Thank you.
Thanks. Hi, Alex. It's Jasper Slingsby. Nice to see you up on screen.
I can't see you, but I'm so sorry I didn't get to see you in person. I hope that we do in 4 years.
Don't worry. Much good wine was drunk by all, so yeah, I'm afraid you missed out.
Sure. I know. I'm so sorry.
Yeah, great talk. I have to say, I'm always petrified when I see the type conversion in chaparral with the alien grasses. And it's something that fascinates me because we don't really get that here to the same extent. So you have sites, we have all the same grasses, they are far more abundant than we would like, but they essentially only occur where there's been severe disturbance of the soil. And in our nature reserves, they only really seem to get a foothold in sites where we have essentially plowing by feral pigs that kind of just really disturb large patches of the soil. And I know feral pigs are a big problem in California, but I struggle to believe that there'd be enough of a problem to convert that much chaparral. So I'm curious as to why chaparral is so vulnerable to it, whereas our system seems really resistant to it somehow. I don't know if you have any thoughts on that.
Yeah, I mean, I don't know the ecology there as well, but I mean, one of the problems is just simply the frequency of fire. In that I don't know what the fire return intervals are in the areas you're talking about, but we have a fairly high proportion of obligate cedars, which are the species that are most vulnerable to it. And that is, it prevents the adequate recovery of species. And the grasses are so pervasive and they're so prevalent, they're so highly dispersed that they basically will colonize any opening in the landscape. We also have a legacy of grazing in Southern California. So this type conversion has actually been going on for quite a while because there had been purposeful type conversion to convert a lot of the land to grazing. So it could be that there's just a greater, sort of a more prevalent distribution of grasses as a source of dispersal into the disturbed areas.
Cool, thanks. I'll let others ask a question.
So Alex, I'm going to answer that question real quick. This is Hugh. So I think, Jasper, a lot of it has to do with soil fertility. A lot of it. Yeah, because if you look at it was interesting having Kathleen's talk before this, because on serpentine soils, we don't get grass invasion after fire.
Right.
And we just don't have huge areas of these really poor, quartzite sort of sourced soils that you've got here. And my guess is that has a lot to do with it. Anyway, it's my 2 cents.
Yeah, that's a good point.
Hey, Alex.
Yeah, the other thing I was going to say is that there's also been a lot of conversion of our sage scrub, soft chaparral species to grass. And in certain areas, there's been a hypothesized link between nitrogen fertilization and that favoring the grasses in those areas.
Hi there. I think we're also dealing with a new normal in the Western Cape of South Africa, seeing more fires in winter and our fire seasons being a lot longer. I'm not sure whether we're having larger fires, but we are having higher frequency of fires. But then we do have sort of really bad years. I think maybe just a comment on firefighting itself. We spend a lot of time protecting structures when we should actually be protecting the felt and keeping those fires to as smaller a size as possible. And I see one of your slides, you had something like 18,000 structures. And our big one fire in Eisner in 2017, we had about 1,000 structures. So it's an issue that we are spending a lot of time putting ourselves at risk and protecting infrastructure when we should actually be protecting the felt. And maybe your comments on that, how you deal with it in the States.
Yeah, it's a good point. And one of the big foci of my research has been the human influence on fire, particularly related to development patterns and the arrangement and location of housing development, because that influences ignitions. It also influences where structures are most likely to be destroyed, as well as where firefighters need to get to be able to protect structures. And in California, of course, we've had massive expansion of the wildland-urban interface, and right, it's the almost entirely the firefighting is allocated toward structure defense, and it's not really, there's really not a paradigm here of firefighting to prevent fires from entering ecologically sensitive areas. Although, I take that back. We have been working to conserve or to save the valuable sequoia trees in recent fires. But for the most part, it is about structure protection. And, I think one of the lessons that I typically talk about to other Mediterranean ecosystems is to not follow in California's footsteps in terms of the way you develop into the landscape, because that really stretches out firefighting resources. And a lot of times when the worst fires break out, it is during severe fire weather conditions where there's multiple fires breaking out at the same time. And so that even further diminishes the capacity of firefighters to be able to do the work they need to do, because there's multiple destructive fires happening at the same time.
Thank you. Do we have any other? We have time for one more question. Am I missing anybody? All right. Alexandra, thank you so much for sticking it out. Apologies again for that delay. But
No
we
worries.
really do appreciate you joining us and for that amazing talk. Thank you so much. Can we give her another round? All right. I'm going to hand over to Martina.
Hi, everyone. Thank you so much for staying until the bitter end. Really appreciate it. And they say save the best for last. So I think those talks were really interesting. Maybe that kept you here. We're going to quickly do a round of just thanks and prizes, just to close off sort of some of the formalities. If you're a custodian member of the FYNBOS Forum and you're in the room, can I ask that you come forward and sort of come and stand around me, if you don't mind.
It's not so bitter.
They're just bringing some of the gifts forward, so we'll just give them a little minute. Okay, thank you everyone. It really was great to meet, I think, almost all of you, and to have you here for the full 5 days. It was my first Medecos. I've been attending a few FYNBOS forums, but this one has been a really enlightening experience, and we hope to see you at the 2026 conference, which we hope will be in Australia. And I think we mentioned that it's looking like it will be Perth as a destination. So see you in 4 years, hopefully. I think I'd first like to thank Karen, Jasper, and Adrian specifically for helping with populating a really exciting scientific program. I think if we didn't have that substance, we wouldn't have enjoyed it so much. And it is a lot of information, but remember, everything is available afterwards on the conference app, and we keep it there for a little while. If we move it, we'll definitely let all of you know. But thank you, Jasper, Karen. It goes without saying that, we are a non-profit organization as the FYNBOS Forum, and we didn't have the financial resources, in our bank account to host a conference of this scale. So thank you very much to our prize sponsors, in particular up on the slide there, but also to our funders. The NRF has made substantial contributions so that we can, host something like this, as well as Sayon, FYNBOS Node, Botanical Society, Table Mountain Fund. I hope I'm not forgetting anyone. But yeah, and also, of course, thank you to you for registering and paying, because that is the name of the game. So I think give yourself also a round of applause. And then to our conference organizer, Madeleine Wetlink, your company and your people, Robin, Tina, Stephanie, Hanel, they're all standing there at the back, always smiling, always friendly. And then Prosper and Isabel is also, I think, noteworthy of mention. Prosper is sort of responsible for all the financial things that maybe some of you had to deal with. So I believe many of you have been in contact with Prosper. There he is. And Isabel, our online sort of coordinator. Thank you very much. I think we had smooth sailing with the technical staff that we also had. Adrian, you and your team from Audio Pimps, and Charles, Stream Team, James, Daniel. Maybe I'm forgetting one or two people, but a huge round of applause to you guys also for your companies. Okay? David. And then there's a couple of keynote speakers that we haven't formally thanked. I know Wendy Foden might have left, but I thanked Wendy a bit earlier today. And then Kirsten, Frank, please come forward so that we can also just, give you a hug and shake your hand and say thank you for the contributions. Yaku Leroux, I thanked him this morning. He also had to leave a bit earlier, so thank you, Yaku. And especially to Rob, Kathleen, Alexandra, and Mingjen for their online presentations also. Yeah, stay here, please. Lastly, session chairs, Mashudu and Donald. You guys have been very busy. I could always call on you if there was a little glitch or something, so thank you so much. As well as Stuart, Carla, Anina, Shirk, Kirsten, and Davidine for chairing different sessions. That's also not an easy job, so thank you so much to you guys. The posters were printed out for everyone, but you're very welcome to take your poster with you if you contributed a poster. Otherwise, we will repurpose it perhaps at, I think, Kirstenbosch or some educational venue where we can put it up. So if it's difficult for you to travel back with your poster, then you can just let us know. And then I think the last formality is to hand over to Kirsten and Carla. Are we still doing spot prizes also? Yes. Okay. Should I say something about it? Oh, okay. So you can still qualify for spot prizes or our lucky draw. So if you want to quickly write your name, if you haven't done it, no double entries, then please pop your name in that bag with Adrian quickly.
Or you can put your name tag in. Kill two birds with one stone. You can throw your name tag in. Well done.
But so I'm going to hand to Carla and Kirsten to do the prizes based on the evaluations that were also going on in the background. So thank you to you two also for doing that, and Donald and Mashudu who helped coordinate that.
And while we all sort of get ourselves together, I think from the whole forum, from Medecos, from all of us, a very, very special mention needs to go to Martina for really being our queen bee. And for holding it all together and really pulling together an amazing conference. Without you we couldn't do it. And also to Adrian, your wingman. There we go. Really through the year, as Martina said, this group of people don't get paid, so this is all over and above, and this is your second day job that you do at night. We really appreciate it. Thank you.
Thank you.
All right, so I'm going to quickly, I know you're all desperate to get out for lunch, so we're going to quickly read out our prize winners. So This category is for best poster and runner-up. So our runner-up for best poster is Tamsankwa Msweli. So if you're still here, please come up. For the photo. And our best poster goes to Kirsten Shaw. Has she left? Okay. Okay. Okay, perfect. Okay, so she has left apparently, but we will. Oh, wow, okay, to go to her study site in the Eastern Cape. Good dedication there. Ah. I think we've all lost the plot by this time of the conference. Okay, best student presentation. So our student runner-up, and I'm going to use your colloquial name, the South African easier one, Maria Kolokwa. I did see her fairly recently. Yeah. Okay, we'll see. Yeah. Oh, there we go. Yeah. Yeah, we found her. Yes. I just butchered your name. You were. Yeah. I just. There we go. Well done. I didn't. Yeah. And our best student prize goes to Answa de Villiers. Is Answa still here? Is she? Okay. Okay, Answa has also left. Okay. Thanks, Karen. Okay. Now drink her wine. Don't tell her it was in there. Then our final category, our best presentation and runner-up for best presentation. We had a difficult time choosing our best runner-up, so we've got two winners there. So we've got Hugh Safford and we've got Evan Mayer. Yeah.
Take it on behalf of you. I don't have any luggage to take this home. Actually, I do. I got insurance. Who's driving?
Yeah. And then it was pretty unanimous. Our best presentation goes to Hayley Cawthra. Apparently she has left. Yeah, it was actually quite a difficult decision to make. It did take us a whole hour of trying to collate these things. That just shows the caliber of presentations at this conference. So thanks, everyone. And then I will let Martina end off. Oh, sorry, yes, spot prizes. Sorry, I promised you spot prizes. Okay, you can grab. Yeah.
Sorry.
There? Yeah. Okay. Irene Diodero. Come and grab a book. Until we run out there. Yeah. Okay, yes, yeah. Landi Ratif. And we'll let you choose. You can choose a plant. Thanks to Vula for the plants. No, she got. She already got a prize. Sierra Standish. Yeah. I also got Pride.
We're going to speed it up a little bit because there's quite a lot of them.
Pippa, I can't read your surname. Yes.
Harov.
There we go. Pippa Harov. Caitlin Smith. There we go. Glory. Also can't read your surname. Odo. Sorry. Julia Gagunde. Annabelle Cardoso. Take the plants, yeah. Hopefully. Who's that? Lola. That was a guess. Alvaro. Shoot, the plants are, yeah, that maybe they can't. Yeah. Frank Scher. Ruth Mary Fisher. Anna Agua, I think. Apologies. Adam Wilson. Alexandria Thompson. Okay, let's just see how far we. Thank you. Already got something?
Which invasive are you taking over? Alex, which invasive are you taking over?
Tabang. Hang on, how many plants do we have left there? Alan Wood. I don't know. I just want to see how many plants we have left here. Who hasn't, how many? 1, 2, 3 at least? 1, 2, 3, 4, 5, 6, 7. And 4, 2. And we got maybe 1 or 2 more. Maybe do 2. Maybe do 2 and then we. I just can't tell who's. Liz. You can take a plant, Liz.
Thank you, Carla. Thanks so much. Yeah, if you got a plant now and you have a flight to take home, maybe swap with a local.
Okay, just calm down.
Okay, we're almost there. Last one is just there's a conference evaluation link on the app. So if you liked our conference, please give us some feedback there. Or if you didn't like it, then that's also welcome. And then the prize winners, especially the student categories and the best talks, if you could just come for a photo afterwards, that would be nice. And then finally, thank you so much to the custodians for keeping the fort for the FYNBOS Forum. We haven't thanked you enough, so thank you very much for the time that you always invest in the FYNBOS Forum as an organization also. From my side, and I think from the entire custodian membership, and also Adrian, enjoy the rest of the weekend. Travel safe. South African roads are not always the best, and we hope to see you in 4 years in Australia.
Also, just before everybody runs off, just want to say again, thank you very much to Martina for the unbelievable amount of work that she's put into organizing this amazing conference. She's done so much behind the scenes, guys. So, Martina, thank you very much.
It's off. Sorry, you didn't announce where the next Medecos is happening, who is hosting it, and when.
We don't know yet, but it's going to be in Australia.